and insightful experience as we delve deeper into our session and our topics. I believe with each passing days, our collective knowledge grows and our connection strengthens. Namaste and good morning. I'm Kusum Sahi and has been serving in the Nepal Health Research Council since 2021 in the various disciplines, nutrition, COVID-19 and air pollution as such. This session centers on infectious disease and biomedical research. Before we proceed with our agenda for today, it is with great pleasure that I like to introduce our esteemed chairs for this session. Our first chair is Professor Dr. Jeevan B. Sherchand. Can we please have a round of applause for Professor Dr. Jeevan sir? And I would like to request you to please be on the stage. Thank you very much. Professor Dr. Jeevan sir is a distinguished public health professional with over 2 decades of extensive experience in managing and overseeing critical public health programs in Nepal, with a specialization in combating HIV, tuberculosis, and malaria. Serving as the coordinator of the Ethical Review Board at the Nepal Health Research Council, he has published more than 200 papers. Welcome, sir. Now, I would like to draw your attention to our next chair, Dr. Ashish Shrestha. Can we please give him a round of applause? Dr. Ashish Shrestha is an associate professor at the Department of General Practice and Emergency Medicine at Patna Academy of Health Science. With a master's degree in emergency disaster management, Dr. Shrestha is a dedicated member of the medical community, holding life membership in the Nepal Medical Association and the General Practice and Emergency Medicine Association of Nepal. Let us embrace the opportunity to engage, learn, and collaborate. Together, we have the power to inspire change, drive innovation, and shape the future of our field. I now like to hand over the floor to our esteemed chairs, Professor Dr. Jeevan Sherchand and Dr. Ashish Shrestha. Chairs, the floor is yours. Thank you very much.
Thank you, madam. Okay, so now I think we have already behind the time. So today we have around 9 presenters. So I think each of you is provided at least 10 minutes for the presentation. So I hope that you will present within a time. So now I would like to hand over this mic to my colleagues, Dr. Ashish Shrestha.
Thank you, sir. Very warm good morning. So without delay, we'd like to start the session. Our first speaker is Professor Dr. Prakash Ghimbire. Professor Dr. Prakash Ghimbire is a distinguished professor of microbiology at Tribhuvan University in Nepal, posting an illustrations of 25-year career dedicated to infectious disease control and research. He holds PhD in microbiology from All India Institute of Medical Science. So first speaker, Professor Dr. Prakash Ghimbire, sir.
Cheers, respected delegates. Very good morning. The topics which is allocated to me is infectious disease and antimicrobial resistance, a combined challenge to Nepal's health system. as we all are aware, we are discussing on infectious disease, which is a concern related to UN SDG 3, which clearly says that our commitment to end epidemics of AIDS, tuberculosis, malaria, NTD, waterborne disease, hepatitis, and other communicable disease. As of now, we are still struggling with this. If we see where we stand, what causes the most deaths, comparison of last 10 years between 2009 and 2019, many of the non-communicable disease has stopped, but still we have challenges of communicable diseases like tuberculosis, diarrheal disease, and many others. Where we have progressed on deaths and disability is we have progressed a lot on communicable disease. Still, we are at a point where we have dual challenges of communicable and non-communicable diseases. And malnutrition, air pollution, tobacco tops the list, while we still have diseases which still needs elimination and eradication. When we talk of communicable disease, we could see that the COVID-19 have changed our life expectancy. If we come to Nepal, the life expectancy due to COVID has decreased by 2.9 years, which is a backstrap. If we see influenza still circulating, if we see COVID still circulating, if we see HIV, we have done lots of progress, but still we are not yet there in achieving 95, 95, 95. Although we have expanded our services to reach to everybody who is on target. On coming to tuberculosis, we see that it is still a big problem with unreached people who request diagnosis and treatment. If we see a real picture, we still have MDR cases and extended drug resistance tuberculosis. And this is more often in the area where the population concentration is high towards the south of the country. When we talk of malaria, if we see long back, we used to have about 3.4 million cases and 38,000+ deaths. We have progressed a lot. But if we see, we are not yet there. And where we are is now the scenario is reversed. 93% is imported case and about 7% is indigenous. And if we see the changes in where it concentrates on southwestern part of the country with few foci where we need to concentrate our efforts in elimination. Coming to visceral leishmaniasis, which the country has committed for elimination, we haven't reached to that point, although number of cases have come to hundreds, 245+. Everybody is aware of dengue. Lots of dengue cases these years. Last few years, it's hue and cry. And if we see, it's spread all over the country. Coming to Japanese encephalitis, with the introduction of vaccine, the cases have gone down, but it's still not zero. When we see cholera, there are still cases of cholera even in parts of Kathmandu, which has been reported in last couple of years, meaning that this deadly disease is still there. If we see lymphatic filariasis, which is where country has committed for elimination, we have not yet achieved where we target. Although we have done lots of mass drug administration, we have scaled up but not yet reached. When we come to leprosy, there are many leprosy papers in coming presentations. Still, there are some of the districts, subnational cases are detected, and there are challenges. When we talk of scrub typhus, there are cases. Still, we need to work more. And very dreadly is rabies, which is still killing people. We haven't achieved that. When we talk of COVID, we all are aware, huge number of cases in our countries and few thousands of deaths. We all are aware there are measles cases. We did immunize and we did provided second opportunity for measles immunization. Still, in last couple of years, there were outbreaks even in some part of the country. The second part of my discussion is antimicrobial resistance. We all are aware when Alexander Fleming discovered antibiotics, he alerted all of us to use it rationally. The day will come when we will have nothing to treat. And we still have these perceptions, like usual, I know antibiotics can cure everything quickly, while unauthorized prescribers do prescribe on a different manner. All fevers requires injections followed by colorful capsules, and even GP practice empirically. And there are full course which cost a lot, and economically there are people who cannot buy the full course at one go, and it's difficult. The challenge is still there are many cases and many deaths due to AMR. It's difficult to quantify because our health system data is not targeted towards AMR and deaths related to AMR. But some of the studies has clearly shown that there are 6,400 deaths and 23,200 deaths associated with AMR in 2019. When we talk of this, there are directly AMR associated and indirectly associated. There are few organisms which we know, but still there are many others which we may not know. There are few studies which have found out there are few antibiotics which are commonly prescribed in spite of different scenario, different protocols in different hospitals in different years. But dispensing and prescription are different. If we see there is no collaboration, and there is resistance to few organisms, the resistance developed by few organisms, if we see that it's very difficult, and the prevalence of MDR is increasing, and it's challenging. It's challenging in the way that the common antibiotics are almost resistant, and the protocols which are used by physicians are at times challenged because the empirical treatment may not work, and last resort antibiotics are also developing resistance. And if we see that almost 30% in this study which evaluated different studies are resistant, meaning that we are treating only 70%. So 30% are ignored. Even here, 70 prevalence of these isolates in 70% among the included 5 studies, meaning that there is resistance going hayward. And the common antibiotics are getting resistance to those bugs, bacteria. So who is contributing to all these? We all are responsible, whether this is a laboratorian or prescriber or a receiver. We all are responsible. The take-home message on all this discussion is infectious disease still causes significant morbidity and mortality. Preventive measures are important to control these diseases. Continuous efforts are essential to control the disease using effective measures. Effectiveness of programs may be increased through maximizing synergies between programs and health system governance. Multi-disease elimination requires multi-sectoral approach, not only one, to address social, economic, behavioral determinants of health. And antimicrobial resistance is a public health threat with increased burden to the health system, requiring effective antibiotic stewardship measures for timely containment. It looks like it's difficult. We have dual problem or even more, multifaceted problem, communicable disease, non-communicable disease, antimicrobial resistance. And although it looks difficult to walk down to that beautiful mountain, if you keep walking with good governance, we could reach to the target very easily. Thank you. Over to you. Thank you.
Thank you very much, sir. So we will take all the questions at the end of the session. So thank you very much, Professor Dr. Prakash Ghimire, for highlighting the burden of infectious disease and threat of antimicrobial resistance. So I would like to move on to the next presenter. So the next presenter is Divya Rana, who holds Master of Science degree in molecular biology from China Medical University, and currently serving as a senior research officer at the Microbacterial Research Laboratory located at Anandaban Hospital in Lalitpur district since February 2018.
Respected chairs and seniors and dear delegates and audience, thank you for providing me the opportunity to present research from our laboratory. I belong to the Microbacter Research Laboratory, which is part of the Anandaban Hospital, which is the biggest referral hospital for leprosy cases in Nepal, and everything is free for the leprosy cases there. And Anandaban Hospital is in turn part of the Leprosy Mission Nepal. So I am going to present about the association of a human leukocyte antigen HLA-V 1301 and dapsone-induced hypersensitivity reaction in Nepalese leprosy patients. Sorry, okay. म चाहिँ नेपालीमा यो प्रेजेन्टेसन दिन कोसिस गर्दैछु है, अलि बुझियोस् भनेर हैन। हामी चाहिँ धेरै जसो चाहिँ कुष्ठरोगलाई चाहिँ माइकोब्याक्टम लेप्रेसँग जोड्छौँ, तर 2008 देखि चाहिँ यो माइकोब्याक्टम लेप्रोम्याटिसिसले पनि कज गर्छ भनेर चाहिँ रिसर्चहरू आइरहेको छ। केही वर्ष गएको केही वर्षहरूमा अहिले चाहिँ नेपालमा 2000 देखि 3000 केसहरू प्रति वर्ष आइरहेको छ, र यसलाई चाहिँ WHO ले रेकमेन्ड गरेको मल्टिड्रग थेरापीले ट्रिट गरिन्छ। मल्टिड्रग थेरापीमा चाहिँ रिफाम्पिसिन, क्लोफाजिबिन र ड्याप्सन हुन्छ, र ड्याप्सन चाहिँ सबभन्दा पुरानो ड्रग हो। यो चाहिँ 1940s देखि चाहिँ कुष्ठरोगको लागि चाहिँ युजमा आइरहेको छ। सबै ड्रगहरूको चाहिँ एडभर्स ड्रग रिएक्सन अथवा साधारण भाषामा भन्दाखेरि चाहिँ साइड इफेक्टहरू हुन्छ, र यसले चाहिँ केही संस्थाहरूले भनेको छ कि आउट पेसेन्ट्सहरूमा 25% अफ द केस चाहिँ एडभर्स ड्रग रिएक्सनको कारण हुनसक्छ भनिन्छ। एडभर्स ड्रग रिएक्सन अथवा साइड इफेक्ट्सहरू चाहिँ दुई किसिमको हुन्छ। एउटा चाहिँ युजुअल टाइप 1 भनिन्छ, जुन चाहिँ हामीले चाहिँ गेस गर्न सक्छौँ, जस्तो कुनै औषधि खाएर हामीलाई डिजिनेस हुन्छ, ग्यास्ट्रिक हुन्छ, रिङ्गिटा लाग्ने हुनसक्छ, र त्यो चाहिँ 80% जति चाहिँ साइड इफेक्ट्सहरू चाहिँ त्यो हुन्छन्, र केही ड्रगहरू एडभर्स इफेक्ट रिएक्सन्सहरू चाहिँ अफ टार्गेट अथवा चाहिँ डक्टरलाई थाहा हुँदैन कसलाई चाहिँ यो एडभर्स रिएक्सन हुन्छ भनेर हैन, र यसलाई चाहिँ मेडिकल टेक्स्ट बुकहरूमा चाहिँ इडियोसिन्क्रेटिक भन्ने गरिन्छ। इडियोसिन्क्रेटिक भनेको चाहिँ जसको चाहिँ रिजन हामीलाई थाहा छैन, कसलाई हुन्छ हामीलाई थाहा हुँदैन। र म अहिले चाहिँ ड्याप्सन हाइपर सेन्सिटिभिटी मतलब कुष्ठरोगको 3वटा ड्रग मध्ये एउटा ड्रगको हाइपर सेन्सिटिभिटी रिएक्सन को बारेमा भन्दा गइरहेको छु, र त्यो चाहिँ टाइप 2 हो। टाइप 2 हो र त्यो चाहिँ ड्याप्सनसँग रिलेटेड छ। यो फोटो चाहिँ एउटा मान्छेलाई ड्रग एलर्जी भएको 2 महिना पछि लिएको फोटो हो। यसको मतलब सोच्नु कि सुरुमा कस्तो भयो होला। र ड्रग एलर्जी भन्दाखेरि चाहिँ हामीले चाहिँ अलिकति के कन्फ्युज हुन्छ भने ड्रग एलर्जी भनेपछि कहिलेकाहीँ हामी पेनिसिलिन एलर्जी अथवा कहिलेकाहीँ चिलाउने एलर्जीसँग सम्बन्ध राख्छौँ, सोध्छौँ, तर त्यो चाहिँ होइन। अलिकति यो ड्रग एलर्जी भनेको चाहिँ एकदम सिरियस एलर्जी हो। यसलाई हामीले अर्को भाषामा चाहिँ सिभियर क्युटेनियस एडभर्स रिएक्सन भन्छौँ। यो ड्रग एलर्जी सुरु हुनको लागि युजली चाहिँ 30 दिन 1 महिना औषधि खानुपर्छ हैन। मतलब एकै दिनमा एलर्जी हुँदैन, 1 महिना औषधि खानुपर्छ युजली। र 1 महिना औषधि खाएर ड्रग एलर्जी सुरु भयो भने अनि त्यो औषधि खान रोक्यो भने पनि त्यो सन्चो हुँदैन। आउने 1 हप्तादेखि 3 महिनासम्म पेसेन्ट्सहरूलाई चाहिँ हस्पिटलमा बस्नुपर्ने हुन्छ। र कसै कसैको अहिलेसम्मको डेटा अनुसार नेपालमा चाहिँ 10% को मृत्यु हुन्छ। एकदम कडा किसिमको एलर्जी हो। र अहिलेसम्मको यो मेटा एनालिसिसबाट के थाहा पाएको छ भने डिफ्रेन्ट देशहरूमा हैन, जस्तो अहिलेसम्म चाइना, थाइल्यान्ड, ताइवान, साउथ कोरिया र इन्डिया अनि इन्डोनेसियामा पनि गरेको स्टडीहरूले चाहिँ यो HLA-V 1301 भएको मान्छेहरूले चाहिँ ड्रग यो ड्याप्सन खायो भने उनीहरूलाई एलर्जी हुन्छ भनेर देखाइरहेको छ। र मलाई पनि लाग्यो कि अब यो टेस्ट भइसकेको छ, रिजल्ट आइसकेको छ, नेपालमा किन गर्ने भनेर। तर हाम्रो डाक्टर साहब हुनुहुन्छ, डाक्टर महेश शाह, उहाँ चाहिँ मेडिकल डाइरेक्टर हुन्छ र सिनियर डर्माटोलोजिस्ट हुन्छ। उहाँले भन्नुभयो कि किनभने एथनिसिटीले पनि डिफ्रेन्स ल्याउँछ हैन। के थाहा हाम्रो नेपाली पपुलेसनमा चाहिँ यो HLA-V 1301 छँदै छैन, त्यसैले चाहिँ हामीले नेपाली पपुलेसनमा पनि टेस्ट गर्नुपर्छ भनेर उहाँले भनिसकेपछि चाहिँ हामी यसमा चाहिँ अगाडि बढ्यौँ र हामीले यो स्टडी सुरु गऱ्यौँ। हुन त यो चाहिँ 2018 मा फन्ड भएको थियो, तर केही कारणले र कोभिडले गर्दाखेरि पनि हामीले यसलाई सुरु गर्न सकेनौँ र जुन 2022 मा सुरु भयो। यसमा हामीले 2 किसिमको केसहरू लिइरहेको छौँ, रेट्रोस्पेक्टिभ केस र प्रोस्पेक्टिभ केस। रेट्रोस्पेक्टिभ केस किन लियो भनेपछि मतलब कसैलाई एलर्जी भइसकेको छ र ऊ बाँचेको छ भने ऊ कुष्ठरोगी पेसेन्ट भएको हुनाले ऊ चाहिँ हाम्रो हस्पिटलमा चाहिँ आइरहन्छन् र उनीहरूलाई हामीले उनीहरूको थोरै रगत लिएर यसमा चाहिँ त्यो जिनको टेस्ट गऱ्यौँ। त्यो चाहिँ एसोसिएसन स्टडी थियो हैन, जसले चाहिँ हामीलाई हेल्प गर्छ कि त्यो HLA-V 1301 थियो कि थिएन जसलाई चाहिँ एलर्जी भयो। अनि त्यसमा चाहिँ हामीले ड्याप्सन टोलरेन्ट अथवा कन्ट्रोल केस पनि लियौँ, जसले चाहिँ कमसेकम 3 महिना ड्याप्सन खाँदा पनि कुनै किसिमको एलर्जी नभएकोलाई चाहिँ हामीले केस मान्यौँ। र सिमिलरली चाहिँ प्रोस्पेक्टिभ केसमा चाहिँ के गर्यो भनेपछि जुन 2022 पछि चाहिँ जुन 2022 पछि चाहिँ हामीले चाहिँ सबै न्यू केसेसहरूमा हाम्रो हस्पिटलमा आउने सबै न्यू केसहरूमा चाहिँ हामीले चाहिँ यो HLA टेस्ट गऱ्यौँ र जसमा HLA-V 1301 छ उसलाई ड्याप्सन हामीले दिएनौँ। र यसले हेऱ्यौँ कि HLA-V 1301 टेस्टले चाहिँ एलर्जी हुनबाट जोगाउँछ कि जोगाउँदैन, अथवा चाहिँ उनीहरूको हामीले ज्यान पनि जोगाउन सक्थ्यौँ। यो हाम्रो मेथड्स हो हैन। यसमा चाहिँ हामीले चाहिँ एउटा भ्यालिडेटेड QPCR मेथड प्रयोग गरेको छौँ। यो चाहिँ हाम्रो हेड अफ अहिले दिनुहुन्छ, उहाँले हेड अफ डिपार्टमेन्ट हाम्रो कान्छा दाइले कलेक्ट गरेको डेटा हैन। 2000 देखि 2021 सम्म हाम्रो पेसेन्ट्सहरूमा ड्रग एलर्जीको प्रिभिलेन्स कति छ हैन। यो चाहिँ 2% देखि 3% को बिचमा छ। यो चाहिँ रिजल्ट पेज हाम्रो सुरु भयो जुन 2022 देखि फेब्रुअरीको इन्डसम्म हामीले 287 न्यू केसहरू इनरोल गरिसकेको छौँ। र त्यसभरिमा हामीले हेर्दाखेरि थाहा हुन्छ ग्रामीण क्षेत्रीहरू सबभन्दा बढ्ता छन्, फलोड बाई जनजाति, अनि त्यसपछि मधेसीज, अनि त्यसपछि नेवारी। हामीले चाहिँ जनजाति र नेवारीलाई चाहिँ डिफ्रेन्सिएट गर्दैछौँ किनभने हाम्रो चाहिँ नेपालमा जनजाति भनेको चाहिँ सोसियो इकोनोमिक क्लासिफिकेसन हो हैन, यो चाहिँ एथनिक क्लासिफिकेसन होइन। र हामीले चाहिँ डेटा चाहिँ डिफ्रेन्ट एथनिसिटीमा चाहिँ HLA-V 1301 को प्रिभिलेन्स पत्ता लाग्योस् भनेर चाहिँ छुट्याएको छौँ। यो यसरी हामीले हेर्दाखेरि चाहिँ हुन त यो स्याम्पल साइज सानो छ, थारूहरूमा चाहिँ 40% मा चाहिँ HLA-V 1301 छ हैन, यो एकदम उ छ हैन। र त्यसपछि चाहिँ जनजाति मुस्लिम्सहरूमा, तर मुस्लिमको पपुलेसन पनि सानो छ, स्याम्पल साइज 4 छ हैन। त्यसपछि जनजातिहरू छन्। र हामीले के पायौँ भनेपछि हाम्रो केसहरूमा चाहिँ जसलाई चाहिँ एलर्जी भएको छ उनीहरूमा चाहिँ 67%हरूमा चाहिँ HLA-V 1301 छ र कन्ट्रोल्सहरूमा पनि छ, तर त्यो थोरै छ, 5% 4% जति छ। र यो चाहिँ हाम्रो अलिकति चाहिँ यो सेन्सिटिभिटी चाहिँ कम भएको हामीले पायौँ हैन। हाम्रो चाहिँ सबै ड्याप्सन एलर्जी केसहरूमध्येमा हामीले 67 मा मात्र यो एलर्जिक यो HLA-V 1301 को प्रेजेन्स पायौँ हैन। र यो चाहिँ हामीले चाहिँ डाक्टर शाहहरू अनि हामीहरूले चाहिँ यो किन यो चाहिँ के यसको बारेमा हामीले सोच्यौँ यो किन हुन सक्छ भनेर, किनभने अनि हामीलाई लाग्छ हैन, अहिले हामीलाई थाहा छैन, हामीलाई लाग्छ कि यो चाहिँ हामी कहाँ कहिलेकाहीँ एलर्जिक पेसेन्ट्सहरू चाहिँ बाहिर पेरिफेरल क्लिनिकबाट चाहिँ यो रिफर भएर आउँछन् र पेरिफेरल क्लिनिकमा यदि एलर्जी लाई राम्ररी चिन्न सकेन भने चाहिँ उनीहरूले यो ड्याप्सन एलर्जी भनेको हुनसक्छ र त्यही डिफिनेसन हामीले मान्नुपर्ने हुन्छ, किनभने हामीले फेरि उनीहरूलाई चाहिँ ड्याप्सन दिन सक्दैनौँ। तर हामीले चाहिँ यो जसलाई यहाँ मैले फल्स नेगेटिभ लेखिरहेको छु र जुन चाहिँ ट्रु पोजिटिभ हो, त्यो दुईटालाई चाहिँ हामीले एउटा चाहिँ एलर्जीको वर्ल्डमा चाहिँ हामीले एउटा रेजी स्कार भन्ने एउटा चाहिँ छ, त्यहाँनिर यहाँ तल मैले यो रेफरेन्स दिइरहेको छु। रेजी स्कार भन्ने चाहिँ एउटा ग्रुपले चाहिँ ड्रेस क्राइटेरियालाई चाहिँ प्रोबाबिलिटी स्केल बनाउँछ, सिभिरिटी स्केल होइन, प्रोबाबिलिटी स्केलको आधारमा हामीले चाहिँ क्लासिफाई गर्यो फल्स नेगेटिभ र ट्रु पोजिटिभहरूलाई। र यसरी हेर्दाखेरि चाहिँ ट्रु पोजिटिभको चाहिँ 92% अफ द पेसेन्ट्स चाहिँ आइदर डिफाइनेट अथवा प्रोबेबल ड्याप्सन एलर्जी केस हो भने मात्र करिब 17% चाहिँ ड्याप्सन एलर्जी केस हुन्छ त्यो स्केल प्रयोग गरेर। त्यसैले सायद जुन चाहिँ जसमा चाहिँ हामीले चाहिँ एचएलभी नेगेटिभ पाइरहेको छौँ त्यो सायद ड्याप्सन एलर्जी थिएन। जस्तै ड्याप्सन एलर्जी जस्तै सिम्टम्सहरू चाहिँ मेट हेमोग्लोबिनेमिया अथवा चाहिँ एनेमियामा पनि देखिन सक्छ। र हामीले चाहिँ पहिलोचोटि चाहिँ एलर्जिक पेसेन्ट्सहरूमा चाहिँ हर्पिस भाइरस हेऱ्यौँ होइन, यो चाहिँ पिसिआर मेथडबाट हर्पिस भाइरस 5 र हर्पिस भाइरस 6 हेऱ्यौँ। यो चाहिँ ड्याप्सन एलर्जीको केसमा यो चाहिँ एकदम नन केस हो, एलर्जीमा चाहिँ हर्पिस भाइरसको इन्फेक्सन हुन्छ भनेर चाहिँ नन फिनोमिना हो, तर ड्याप्सन एलर्जीको केसमा चाहिँ थिएन। त्यसैले हामीसँग चाहिँ अलिकति यो पेसेन्ट हामीले कलेक्ट गर्न सक्यौँ, स्याम्पल कलेक्ट गर्न सक्यौँ र हेर्दाखेरि चाहिँ इन्डिड यसमा पनि ड्याप्सन एलर्जीमा पनि यो हर्पिस भाइरसको इन्फेस्टेसन हामीले देख्यौँ। अहिले चाहिँ हामीले एउटा कमर्सियल किट प्रयोग गरिरहेको छौँ, तर यो पार्ट चाहिँ मेरो एउटा पिएचडी को पार्ट पनि हो होइन। मेरो चाहिँ मेरो सुपरभाइजर डाक्टर जरिना जोशी सेन्ट्रल डिपार्टमेन्ट अफ बायोटेक्नोलोजीमा हुनुहुन्छ र यसमा चाहिँ यसको पार्ट थेसिसको पार्टको स्वरूपमा चाहिँ अलिकति सस्तो होस् भनेर चाहिँ यो चाहिँ डुप्लेक्स पिसिआर हामीले चाहिँ डेभलप गरिरहेको छौँ, जुन चाहिँ सस्तो हुनसक्छ होला भनेर हामीले यो चाहिँ डेभलप गरिरहेको छौँ र तर यो भ्यालिडेसन चाहिँ अहिले हुँदैछ। अर्को चाहिँ हामीले ल्याम्प टेक्निक पनि डेभलप गरिरहेको छौँ। ल्याम्प प्राइमर चाहिँ एउटा फ्री आइकन केमिकल्स भन्ने चाहिँ फ्री वेबसाइट छ जसले चाहिँ हेल्प गर्छ एनसिबी प्राइमर ब्लास्ट जस्तो गरेर, त्यसमा चाहिँ यो काम गरेर चाहिँ यो प्राइमर यो ल्याम्प मेथड पनि मैले यो हेर्दैछु होइन कि जुन चाहिँ सहिलोसँग चाहिँ डायग्नोस गर्न सकियोस् भनेर एचएलभी 1301लाई, तर यसको पनि भ्यालिडेसन हुँदैछ। कन्क्लुजनमा म यो भन्न चाहन्छु कि यो एचएलभी 13 र अलिकति अघि मैले भन्न छुट्याएँ कि यो हाम्रो केसहरूमा चाहिँ ठिकै छ के भन्न छुट्याएँ भनेपछि चाहिँ हामीले चाहिँ यो स्क्रिनिङ सुरु गरिसकेपछि कसैलाई पनि एलर्जी भएन, तर यो दौरानमा हामीसँग चाहिँ करिब 287मा करिब 5 देखि 10 वटा एलर्जी केस भइसक्नु पर्ने थियो हाम्रो हस्पिटलमा। तर एचएलभी 1301 स्क्रिनिङ गरेर एचएलभी 1301 पोजिटिभ पेसेन्ट्सहरूमा चाहिँ हामीले ड्याप्सन नदिँदाखेरि अहिलेसम्म कसैलाई पनि एलर्जी भएको छैन। र त्यसैले चाहिँ हामीले चाहिँ यो इन्स्टिट्युसनलाइजेसन अफ एचएल स्क्रिनिङ बिफोर एडमिनिस्ट्रेसन अफ मल्टिड्रग थेरापी भयो भने चाहिँ हामी चाहिँ यो हेल्पफुल हुन्छ भनेर सोचेको छौँ है। ल थ्याङ्क यू दिन चाहन्छु होइन मेरो चाहिँ सुपरभाइजर्सहरूलाई ल फ्रेन्ड्सहरूलाई थ्याङ्क यू।
Thank you, Mr. Divya. So from this presentation, we'll be taking 2 questions. But for the first presentation, we'll be taking questions at the end. So if anyone has any question, please raise your hand.
Because you have to answer the questions. So you sit there. Okay. Nasa, please. Raise your question. Microphone.
Thanks for nice presentation. I am Dr. G.D. Thakur, ex-director. I have worked several years for the leprosy, and thanks for your institution. I have visited so many times. I am very impressed with your research, and that will give the direction to the leprosy patients. My question is, how you should have to translate one data you have shown there. Every year we are getting there 2,300 new patients, and since long time we are getting the same data. Near about 13 years ago, that was the same data. So what's your idea how you should have to reduce the case? and how we should have to maintain the leprosy elimination at the district level. National level you have achieved in the 2010, but yes, still we have not achieved at the district level. So that's two questions I have. Thank you.
थ्याङ्क यू भेरी मच सर म हजुरलाई चिन्छु सर होइन अनि म चाहिँ ल्याबको मान्छे भएको हुनाले सर मलाई यसको धेरै एन्सर त थाहा छैन तर हामीले चाहिँ लिटरेचरमा पढे अनुसार चाहिँ यो चाहिँ कन्स्ट्यान्ट हुनुको कारण चाहिँ यो चाहिँ इनगोइङ ट्रान्समिसन हो होइन अनगोइङ ट्रान्समिसन हो अब अनि युजुअली चाहिँ अहिले चाहिँ हामीहरू चाहिँ इनएक्टिभ अथवा प्यासिभ केस डिटेक्सनमा छौँ जसमा चाहिँ हामी चाहिँ पेसेन्ट्सहरू चाहिँ आफै आओस् र आफ्नो लिजन्सहरू आफ्नो चाहिँ कुष्ठरोग देखाओस् र हामी ट्रिट गर्छौँ भन्ने हामी मेथोलोजीमा छौँ यदि हामी एक्टिभली यदि गयो भने एक्टिभली केस फाइन्डिङमा गयो भने सायद हामीले चाहिँ त्यो कम गर्न सक्छौँ होला म चाहिँ यसको एन्सर चाहिँ राम्रै दिन सक्दिनँ सर
Thank you. Next, Dr. Dwarika would like to ask the next question.
First of all, congratulations for this interesting study. We know, as a dermatologist, I'm from a teaching hospital, how difficult and how severe is the dapsone hypersensitivity. As you have said that nowadays we included it as a DRESS syndrome, and how long it takes. Just that aru deshma pani bhaisakya rahecha haina. If we can, as you said, if we can screen HLA, to your type ko HLA, avoid giving dapsone to the patient, which nowadays we are still giving, that would be a great asset. Just that in this study, control population चाहिँ कुन थियो?
कन्ट्रोल चाहिँ सर 3 महिना लगातार ड्याप्सन खाइसकेको अब धेरै जसो चाहिँ हाम्रो आरएफटी केसेसहरू हैन मतलब 12 महिना औषधि खाइसकेको पेशेन्ट्सहरू जसलाई चाहिँ कुनै किसिमको एलर्जी भएन अथवा तर हाम्रो चाहिँ मिनिमल डिप्रेसन चाहिँ कम्तीमा 3 महिना लगातार ड्याप्सन खाइसकेको तर कुनै किसिमको एलर्जी नदेखिएको पेशेन्ट्सहरू थियो सर
त्यसमा केस र कन्ट्रोल चाहिँ वेल म्याच थियो
थिएन यो मतलब हामीले चाहिँ यो किनभने ड्याप्सन केसेसहरू चाहिँ अब र्यान्डमली जो चाहिँ हामी जस्तो 20 वर्ष अगाडिको ड्याप्सन केसहरू जो चाहिँ आउँछ हैन जो पनि होस् बच्चा होस् अथवा बुढो होस् सबैलाई केस
ठीक छ ओके थ्यांक यू
थ्यांक यू सर
thank you Mr. Nibyarana for the wonderful presentation and I would like to move on to the next presentation. The third speaker is Ganga Gharti Chetri. Ganga Gharti Chetri is a dedicated microbiologist with master's degree in microbiology, currently pursuing her PhD at Central Department of Microbiology, Tribhuvan University. In addition to her academic pursuits, she serves as a lecturer in microbiology department at Trisandar Multiple Campus. Over to you, ma'am.
Namaste and good morning. Thank you, Chair, for the introduction. So I am in the front of you to present my research work entitled as Laboratory and Field Evaluation of Biolabicide Bacillus thuringiensis Isolates of Nepal against Aedes aegypti. Would like to thank the team for their continuous support, and the research was carried out in the Central Department of Microbiology, Tribhuvan University. So as we are aware of the dengue disease, so dengue disease rate is increasing in all urban areas of Nepal. All the 77 districts of Nepal are prone to a dengue disease. We know that Aedes aegypti and Aedes albopictus are the main mosquito vector responsible for transmitting the dengue virus in Nepal. Aedes aegypti is the main vector for transmitting dengue virus till date. So when we look at the dengue control measures, search and destroy campaign is carried in all urban areas of Nepal by environmental management and manipulation, as well as diagnosis and treatment is in process, but the disease rate is increasing in order, as we have seen this graph previously also. in last 2 years, the cases are above 50,000. So there is an urgent need for the new tools to decrease the burden of dengue disease in Nepal. Multiple and safe tools should be implemented to control the dengue vector. Tools like biological control agents, sterile insect techniques, and Wolbachia or other tools should be implemented to control the mosquito vector and also included in the vector control programs. As this Aedes aegypti has a different behavior than the other mosquito vectors. It breeds indoor in the domestic containers, and it's a day biter. So larval source management without emptying water containers is the need for the urban areas. This can be done by using a biological control agent like Bacillus thuringiensis BT, and it's a novel work and a new intervention in Nepal. So the research aim is to isolate a mosquito larvicidal Bacillus thuringiensis BT as a future tool to control mosquito vectors in Nepal and my general objective. Based on that, the specific objective is set as isolation of Bacillus thuringiensis from the soil sample, identification by conventional method, as well as by 16S RNA gene sequencing, as well as screening for mosquitocidal activity among the BT isolate, formulation of biolarvicide, evaluation of biolarvicide against Aedes aegypti in the laboratory condition, as well as in field trial. So to achieve the objective, following method was followed. For first one was isolation and identification. So soil sample was collected from different regions of Nepal, and by acetate selection method, different types of Bacillus species was isolated and further identified as Bacillus thuringiensis by observing the crystal protein produced by the bacteria by Comasi brilliant blue staining technique. Similarly, the larvicidal activity was carried by culturing the bacteria in a nutrient agar plate, and those bacteria which produce only a spherical crystal protein was fed to a mosquito larvae collected from different field, and the mortality of the larva was observed after 24 hours in a room temperature as per the WHO guidelines. Similarly, the quantitative bioassay against laboratory red Aedes aegypti larvae was carried out by formulating the liquid form of an biolarvicide. 6 different concentration of the biolarvicide was fed to the mosquito larvae in 4 replicates in 3 different consecutive days, incubated in the room temperature, and the mortality was observed after 24 hours as per the WHO guidelines. So for that, rearing of Aedes aegypti larvae was carried out in the Central Department of Microbiology, in which a cage was built temporarily. Inside the cage, about greater than 300 adult mosquito of Aedes aegypti were placed in the 3:1 ratio, female is to male ratio, and they were fed with an expired blood plus 10% sugar solution to produce an eggs. The eggs were hatched by dipping in the water, and then the uniform larvae was used for larvicidal activity, as well as the biolarvicide so produced formulated was also evaluated by field trial. Simulated field trial and open field trial was carried out. In case of simulated field trial, small containers were used filled with 1500 ml of water and placed under shaded condition, and different concentration of biolarvicide so formulated was added, and each week 25 laboratory red Aedes larvae were added to determine the residual effect and the efficacy of the biolarvicide. Similarly, in an open field trial at 20 different sites in Kathmandu, different types of container like tires, buckets, top, jar, etc. were placed in the open field as well in the shaded places. In case of an open field, the container was led to fill with the rainwater, and in case of a shaded places, the containers were added manually. Water was added manually, so that it's created a natural environment for the mosquito to come and lay eggs and hatch the eggs into a larva. So after determining the larval density in the containers, different concentration of BT was added to determine the residual effect and the efficacy. So data analysis was done by using Microsoft Excel manually as well, and by Provid analysis to determine the lethal concentration dose 50 and 90, as well as the mortality percentage was calculated as described by WHO. Graph and tables were used to present the data. So the result. So from 454 soil sample collected from different regions of Nepal, we were able to isolate 1,395 Bt isolate, which shows every soil sample contained greater than or equal to different types of Bt isolates. So soil sample is a rich source of Bt. So on larvicidal screening, screening was done by using only a spherical crystal protein Bt organism. So out of 808 Bt, only 4 Bt were found to be an mosquito larvicidal. They were coded as 14P2A, 7P2A, and 8P2A, and 3P1B. So not all the spherical crystal protein were found to be mosquito larvicidal, indicating the specificity of the crystal protein. So the composition matter, not the shape of the crystal protein, or insecticidal crystal proteins. So this is the photograph taken during the study to observe the morphology of an insecticidal crystal protein. You can see the crystal protein, the spherical shape like crystal protein, which can be easily distinguished by observing under a light microscope when the culture is stained with a comassive brilliant blue staining technique. And this is the result of an 16S RNA gene sequencing, which showed that the 3 strains were 99% identical to the control strain, which is recommended by WHO as an biolarvicide to control the mosquito larvae in different kinds of aquatic habitat. That is, Bacillus thuringiensis var israeliensis is the control strain recommended by WHO. So all the 3 isolates are confirmed as an BTI. So this is the phylogenetic tree. By Mega software, the phylogenetic tree was generated, and by neighbor joining method. So the qualitative bioassay shows that greater than 1 parts per million of the concentration of larvicide is required to cause 100% mortality of an Aedes aegypti larvae generally. So by using probit analysis, the lethal concentration dose was calculated, and it was found to be 6.45 parts per million and 37.15 parts per million required to kill 50 and 90% of the larval population respectively. By using the control strain international toxic unit, the potency of the biolarvicide was found to be greater than 16,000 ITU per mg. So the simulated field trial results shows that all the doses, the 6 doses were used, all the doses were able to show 100% mortality of larva within 24 hours, as well as the highest dose, 6 ml per liter of biolarvicide, showed a residual effect for 6 weeks with 100% mortality of the larva. Similarly, in an open field trial, so the different containers were placed in the open field. So in an open field trial, when the tire were exposed to an open field, 2 ml per liter of biolarvicide showed a residual effect for 2 weeks with 100% mortality. But when the tires were kept in a shaded places and the same dose was added, it showed a residual effect for 3 to 4 weeks with 100% mortality. Similarly, in all kinds of domestic containers, the residual effect with 100% mortality was observed for 6 weeks when the containers are inside the shaded places, but in open field, the residual effect decreased to 2 weeks with 100% mortality. So the plastic material or any kind of material doesn't degrade the biolarvicidal activity. So conclusion, the 3 isolates were identified as a mosquito larvicidal bacteria. So laboratory assay and the field trial result proved that the formulated biolarvicide is effective in controlling a mosquito at different breeding habitat. So these 3 isolates can serve as a future strategies for managing the larval source and combating the mosquito-borne illness in Nepal. So takeaway message, so let's combat the dengue disease by using the safe biolarvicide in domestic containers or other aquatic habitat for larval source management to break the transmission chain of dengue virus. So I'd like to acknowledge NHRC for the opportunity to present my research work and the grant. Thank you.
Thank you very much for the wonderful presentation. So in the interest of time, we would like to take 2 questions for the presenter.
Thank you for the presentation. That was wonderful. Myself, Dr. Jeevan Sakhe. मेरो 2टा concern छ यसमा question चाहिँ. के भन्दा तपाईँले लाइभ ब्याक्टेरिया युज गरिराख्नु भएको छ गिभन द्याट द ब्याक्टेरिया आर प्रोन टु लाइक हाम्रो यो जिन ट्रान्सफरले गर्दाखेरि यो ब्याक्टेरियाले भोलिको दिनमा चाहिँ हाम्रो म्युटेसन भएर चाहिँ यो जुन तपाईँले प्रोटिन देखाइरहनु भएको छ इनइफेक्टिभ हुने चान्सेस कति छ र अर्को हामीले लाइभ ब्याक्टेरिया युज गर्दाखेरि यसको इफेक्ट अन हाम्रो एक्वाटिक इकोसिस्टम एन्ड इभेन्चुली टु हामीलाई पनि किन इभेन्चुली त्यो कहीँ कतिबाट त फेरि हाम्रो फुड सिस्टममा आउँछ यो किनभने त्यति बेला त हामीले धेरै नै युज गर्नुपर्छ हामीले यो लाइभ ब्याक्टेरिया त्यसलाई कन्ट्रोल गर्दाखेरि सो त्यो अवस्थामा चाहिँ हामी कसरी कन्ट्रोल गर्ने होला हामीले डेङ्गुलाई त मस्किटोले कन्ट्रोल गर्दाखेरि एक्वाटिक इकोसिस्टम एक ठाउँमा छ किनभने लार्भी आर फेड अपन बाई प्रब्ली सम फिस होइन इभेन्चुली त्यो थ्रुबाट आउँदै गर्दाखेरि त्यसको हेल्थ इम्प्याक्ट चाहिँ के हुनसक्छ फर हाम्रो मान्छेहरूलाई पनि त्यो दुईवटा क्वेसन
So the answer. So first of all, Bacillus thuringiensis is specific. The crystal protein is specific. It is only toxic. There are different varieties of toxin produced by BT. Some of the toxin are to the different insect pest. But you say you BT toxin specifically kill only the mosquito larvae. So it's non-toxic to humans and other aquatic organisms present in the water samples. One, and another one is about the mutation. So when we were going to commercialize, at present I used a mixed culture, means cell, spore and the crystal sub use theme. When we are going to commercialize and when we are going to use in a large scale, definitely the crystal protein has to be purified and to be applied or should be used for the control of mosquito vector. Yes, thank you.
Thank you, madam. Madam, le चिन्नु नै हुन्छ हैन म हामीलाई अब मेरो साथीहरुलाई नि पढाउनु म्याडम धेरै राम्रो काम भएको छ एउटा कुरा चाहिँ मलाई सोध्न मन लाग्यो म्याडम यसमा चाहिँ हजुरले चाहिँ जुन LC50 को कुरा गर्नुहुन्छ 6.45 ppm मा हैन पार्ट्स पर मिलियन मा यसको लागि चाहिँ हजुरले कन्सन्ट्रेसन चाहिँ रहेछ राम्रो लाग्यो र यो चाहिँ कुन चाहिँ टेम्परेचरमा चाहिँ हजुरले युज गर्नुभएको भन्ने कुरा चाहिँ मलाई जान्न मन लागेको एक्चुअलीमा प्रोटिन भनिसकेपछि त हामीले टेम्परेचरको कुराहरु पनि त्यसमा हेर्नुपर्ने हुन्छ कहिले पनि स्टेबिलिटी हेर्नको लागि हैन सो प्लिज
टेम्परेचर सोध्नु भएको हैन सर
हजुर कुन टेम्परेचरमा चाहिँ हाम्रो चाहिँ यो LC50 चाहिँ 6.45 ppm देखाइराखेको छ
इट्स ए रूम टेम्परेचर वी जनरली युज
नर्मली रूम टेम्परेचर
नर्मल रूम टेम्परेचर ओके
थ्यांक यू मैम वन लास्ट क्वेशन यु लाइक टु टेक
थ्यांक यू मलाई साह्रै खुसी लाग्यो बीटी को प्रेजेन्टेसन तपाईले यति राम्रोसँग गर्नु हो कंग्रेचुलेसन र यस क्षेत्रमा डेंगीमा हामी धेरै काम गरेर फर्स्ट टाइम नेपालमा मैले 2006 मा एज ए डाइरेक्टर एस्टाब्लिश गरेको डेंगी नेपालमा छ र यसको कन्ट्रोल मेथोडोलोजीहरुमा जानुपर्छ भयावह पोजिसन आउँछ तपाईले देखाउनु भयो 50,000 केससम्म पुगिसकेछ कन्ट्रोल मेजरमा जाँदाखेरि भूमिका नदिएर दुईटा क्वेशन छ मेरो एउटा अन्य देशमा यो बीटीलाई कसरी युज गरिरहेछ र कति इफेक्टिभ छ एउटा क्वेशन दोस्रो क्वेशन नेपालमा प्रोग्रामले युज गर्दाखेरि कस्ट इफेक्टिभ अहिले वर्तमान जुन मेथोडोलोजीहरु छ डेंगी कन्ट्रोलको त्यसमा बीटी को इन्ट्रोडक्सन कति फिजिबल छ भनेर यो दुईटा क्वेशन छ मेरो थ्यांक यू
Yes, in case of other countries, they are using BT. Sorry, alone, sir.
म चाहिँ पब्लिक हेल्थ को स्टुडेन्ट भएको हुनाले पब्लिक हेल्थ र पोलिसी को कुराहरु गर्न चाहन्छु यसले चाहिँ पब्लिक हेल्थलाई इस्युलाई कसरी एड्रेस गर्छ एक दोस्रो चाहिँ यसले पोलिसीलाई कसरी इम्प्लिकेट गर्छ दुई यो दुईटा कुरा चाहिँ
पोल्युसन
पोलिसीमा
हजुरले दुईटै क्वेशन एकैचोटी आन्सर
सरको क्वेशनबाटै सुरु गरे मैले आन्सरबाटै सो in other countries other countries they have formulated they are using for last 50 years to control the mosquito vector but singly use garirako chaina sir they are integrating with the other control methods just the IVM integrated vector control methods bata tinarलाई utilize gariracha and it's found to be effective integrated gardahari effective ne dekhincha and arko question चाहिँ अब युटिलाइज गर्ने कसरी भन्ने थियो हैन सर नेपालमा पनि यसरी हामीले इन्टिग्रेटेडै गरेर युज गर्दाखेरि चाहिँ इफेक्टिभ हुन्छ बट इन केस अफ जस्तो हामीले अहिले हेरिराखेको चाहिँ मोस्टली सिन इन अ डोमेस्टिक कन्टेनरमा एडिस इजिप्टी इन्डोर भएको डोमेस्टिक कन्टेनरमा भएको हुनाले सिंगलली युज गर्दाखेरि नि इफेक्टिभ देखिन्छ लागिरहेछ मलाई इफेक्टिभली काम गर्छ भनेर लाग्छ बिकज एडिस एबिलिटी इज अ इन्डोर ब्रिडर हाम्रो डोमेस्टिक कन्टेनर वाटर स्टोरिङ कन्टेनरमा भएको हुनाले त्यहाँ हामीले त्यसलाई मात्रै युज गर्दाखेरि पनि इट विल बी इफेक्टिभ एज वी स्टोर वाटर फर अ लङ्गर ड्युरेसन लाइक सिमेन्टेड ट्याङ्क अर ओभरहेड रुफ ट्याङ्कमा गर्दाखेरि चाहिँ इट्स विल बी इफेक्टिभ सो डोज मात्रै मिलाउनु पर्ने देखिन्छ तर यसले इफेक्टिभ देखिन्छ सो बट अरू जस्तो कम्युनिटी पिपल अर इन द आउटडोर इन्भाइरोमेन्टमा युज गर्दा यु हेभ टु इन्टिग्रेट एलोङ विथ द अदर कन्ट्रोल मेथड एन्ड इट विल बी डेफिनेटली बी इफेक्टिभ अनि इन केस अफ यसको पोल्युसनको बारेमा भन्दा दिस पोलिसी सर सो इट्स एन अहिलेको स्टेसनमा हेर्दाखेरि इट सुड बी इन्क्लुडेड इन द भेक्टर कन्ट्रोल प्रोग्राममा इन्क्लुडेड गर्नुपर्छ एज मेनी अफ द कन्ट्रिजहरूले के गरिरहेछ भने दे हेभ अल्सो इन्डिया इन सेन्स VCRC भनेर इन्डियामा नि उनीहरूले गरिरहेछ दे आर अल्सो हेज कमर्सियलाइज्ड and the policy has been made to integrate it in the vector control program. Policy ko kura bhayo sir. अनि अर्को क्वेसनमा चाहिँ इट्स नट इफेक्टिभ जस्तो यिनी बीटीहरूमा चाहिँ भराइटिजहरू छ देयर आर बीटी अर्गानिजम विच क्यान किल द हनी बी जस्तो यो बीटीको फर्स्ट सुरुवातै के थियो इट वाज आइसोलेटेड फ्रम एन इन्फेक्टियस सिल्कवार्मलाई ब्यासिलस थोरेन्जिन्सिस सटो भनेर भन्छ त्यो पहिला आइसोलेटै भएको फ्रम एन युजफुल इन्सेक्ट विच प्रोड्युसेस सिल्क So there are varieties of BT which may be some of the BT crystal protein are toxic to the cancer cell research room. So there are varieties so it does not yo jun maile bhaneko BT israeliensis jun cha so it is toxic only to an mosquito larvae it's non polluted non toxic to other living organisms. That's answer gare sabaiko question.
Thank you very much. So we would like to move on to fourth presenter. Our fourth presenter is Mr. Ashish Shrestha. He is currently the head of medical record in Anandaban Hospital. He served in the hospital for 36 years. He has completed master's degree in economics from Tribhuvan University. Thank you.
Respected chairpersons and co-chair and all delegates, including Dr. Thakur. म चाहिँ प्रेजेन्टेसन चाहिँ सेमी कहिलेकाहीँ नेपाली कहिले चाहिँ इङ्ग्लिसमा गर्छु मैले यहाँ चाहिँ खास चाहिँ मेरो स्टोरी चाहिँ तपाईँहरूलाई चाहिँ यो ललितपुर जिल्लामा चाहिँ लेप्रोसी केसहरू कसरी छ भनेर चाहिँ जस्ट सिनारियो चाहिँ बताउन खोजेको छु यदि हामी त्यो ब्याकग्राउन्डमा हेर्ने हो भने चाहिँ हामीलाई थाहा छ लेप्रोसी इज क्रोनिक यो इन्फेक्सियस डिजिज हो कज बाई माइक्रोब्याक्टेरियम लेप्रे अनि साथीले भन्नुभयो जस्तो अर्को पजिटिभ अर्गानिजम पनि छ यो चाहिँ विशेष स्किन र नर्भमा चाहिँ एफेक्ट गर्छ यदि चाहिँ ढिलै डायग्नोसिस भयो भने मे कज यो डिफर्मिटी र डिसेबिलिटी पनि हुनसक्छ भनेर छ अनि अहिले नेपालमा चाहिँ एलिमिनेसन घोषणा भइसकेको छ यो 2010मा डाक्टर जीत ठाकुरको पालामा अझै हेर्ने हो भने नेपालमा यदि डिस्ट्रिक्ट वाइज हेर्ने हो भने रातो चाहिँ डिस्ट्रिक्टहरू अझै 1 एबोभ 1 पर 10,000 PR छ अनि यो येलो पार्ट चाहिँ हेर्ने हो भने लेस देन 1 पर 10,000 PR छ अनि यो ग्रिन पार्टहरू चाहिँ कुष्ठरोग छैन भनेर चाहिँ रिपोर्टेड गरेको जिल्लाहरू हो हामी चाहिँ ललितपुर जिल्ला चाहिँ यो येलो पार्टमा छौँ भनेको मतलब चाहिँ 1 पर 10,000 लेस देन 1 पर 10,000 मा छौँ अनि अलिकति ललितपुर जिल्लाको कुरा गर्ने हो भने हामीले चाहिँ 1990 देखि 95 सम्म डोर टु डोर सर्वे गऱ्यौँ त्यो चाहिँ धेरै वर्ष अगाडि बिचमा चाहिँ अब हामीले चाहिँ त्यसपछि सर्वेहरू गऱ्यौँ अलिकति चाहिँ हामी यो जस्तै चाहिँ क्याम्पिङहरू स्किन क्याम्प हेल्थ क्याम्पहरू गऱ्यौँ अनि प्लस चाहिँ अहिले आएर चाहिँ हामीले कन्ट्याक्ट ट्रेसिङ पनि गरेका छौँ त्यो बाहेक चाहिँ एक्टिभली चाहिँ यो जिल्लामा पनि अझै जान सकेको छैन यो मेन चाहिँ यो स्टडी गर्नुको चाहिँ उद्देश्य चाहिँ यो हालको सिचुएसन के छ ललितपुर जिल्लामा लेप्रोसीको भन्ने बारेमा चाहिँ यहाँ दर्शाउनु खोजेको हो अनि यो मेथोडोलोजीमा चाहिँ हामीले चाहिँ यो चार्ट रिभ्यू गऱ्यौँ, मेडिकल चार्ट रिभ्यू गऱ्यौँ जो चाहिँ ललितपुर जिल्लाको बिरामी हो। अनि यो चाहिँ आनन्द वनमा गऱ्यौँ। यो चाहिँ 2000देखि 2023 वर्षको चाहिँ डाटाहरू चाहिँ केलाउने काम गऱ्यौँ। त्यसमध्ये चाहिँ हामीले चाहिँ यो भेरिएबल्सहरू छान्यौँ, एज, जेन्डर, अनि यो अनसेट पिरियड भनेर एउटा चाहिँ बिरामीले चाहिँ सिम्पटम नोटिस गरेको दिनदेखि हस्पिटल अथवा हेल्थ इन्स्टिट्युसनमा गएर चाहिँ डायग्नोसिस भएको समयको बिचको दूरीलाई हामीले अनसेट पिरियड भन्यौँ। अनि ब्याक्टेरिया इन्डेक्स, ब्याक्टेरियल इन्डेक्स, अनि WHO क्लास, डिसेबिलिटी ग्रेड हेऱ्यौँ। अनि त्यसमा चाहिँ रियाक्सन डेभलप भएको छ छैन, अनि हाउस होल्ड कन्ट्याक्ट छ छैन इत्यादि कुरा हामी हेऱ्यौँ। अनि यो रिजल्ट चाहिँ हामीले चाहिँ ड्र गर्दाखेरि WHO क्लास यो प्री इलिमिनेसन फेजमा अनि पोस्ट इलिमिनेसन फेजमा चाहिँ के छ भनेर हेरौँ। अनि सुरुको चाहिँ हामीले चाहिँ त्यो फिगर अगाडि हेर्ने हो भने हामीले चाहिँ यो न्यू केस ट्रेन हेऱ्यौँ। 23 वर्षको ट्रेन हेर्दाखेरि चाहिँ इलिमिनेसन अगाडि चाहिँ जिगज्याग अलि बढी चाहिँ जिगज्याग भएर चाहिँ अनि पोस्ट इलिमिनेसनमा चाहिँ बिस्तारै चाहिँ स्लो डाउन भएको छ। अनि यता चाहिँ अर्को चाहिँ यो टेबलमा हेर्ने हो भने चाहिँ WHO क्लास भर्सेस यो पोसिभ्यासिलिटी र मल्टिभ्यासिलिटी यो दुईटा इरामा चाहिँ कम्पेयर गर्दाखेरि टोटलमा चाहिँ प्री इरामा चाहिँ 89 छ, अनि पोस्ट इरामा चाहिँ 57 छ। तर त्यो भित्र हेर्ने हो भने चाहिँ पोस्ट इरामा चाहिँ मल्टिभ्यासिलिटी चाहिँ केसहरू चाहिँ 82% छ, र बाँकी चाहिँ पोसिभ्यासिलिटी चाहिँ 17 छ। हाम्रो चाहिँ स्याम्पल साइज चाहिँ 146 हो। अनि जो चाहिँ कन्फर्म आनन्द वनमा चाहिँ कन्फर्म भएको केसहरू यो चाहिँ 23 वर्षको चाहिँ बिचको डाटा। अनि हामीले चाहिँ यो एज वाइज चाहिँ हामी एज वाइज हेर्दाखेरि लेप्रोसी फिल्डमा चाहिँ बच्चा अथवा चाइल्ड केस भनेको अन्डर 14 अथवा 14 अर लेस देन 14 भन्छौँ। यो चाहिँ हामीले चाहिँ 5% पायौँ। यताको चाहिँ सेक्स रेसियो हेऱ्यौँ भने अथवा चाहिँ यो मेल फिमेल चाहिँ प्रोपोर्सन चाहिँ करिब 55 र 45 छ यो स्याम्पलहरूमा। अनि हामीले चाहिँ अनसेट पिरियड हेऱ्यौँ, बिरामी कति छिटो चाहिँ हेल्थ सिकिङ इच्छा लिएर चाहिँ अथवा चाहिँ मलाई दाग आएको छ, यो चाहिँ लेप्रोसी हुनसक्छ कि सक्दैन भनेर चाहिँ यो खोज्यौँ। एभरेजमा हाम्रो चाहिँ 18 महिना चाहिँ हाम्रो चाहिँ एभरेज लेन्थ अफ यो अनसेट पिरियड रह्यो। त्यसमा चाहिँ अधिकांश 71% चाहिँ विदिन 1 वर्षभित्र नै चाहिँ आएको देखियो। तर 29% स्टिल इन डिले इन डायग्नोसिस है, अझै उनीहरू चाहिँ समयमा आइसकेको छैन। अनि यो ग्रुपलाई हामीले चाहिँ टार्गेट गर्नुपर्छ भन्ने हो। यो ब्याक्टेरिया लोडको हिसाबले चाहिँ हामीले चाहिँ अर्को भेरिएबल हेऱ्यौँ। त्यसमा चाहिँ करिब चाहिँ 37%मा चाहिँ ब्याक्टेरिया पोजिटिभ आयो। एमोङ देम अझ चाहिँ 16%मा त मोर देन 4+ ब्याक्टेरिया भएको चाहिँ हामीले पायौँ। इट मिन्स अझै इन्फेक्सन छ कि भन्ने चाहिँ हामीलाई चाहिँ सजेक्ट गराउँछ। अनि बिरामी त्यो स्टडी पपुलेसनमा हामीले चाहिँ डिसेबिलिटी ग्रेड हेऱ्यौँ, WHO क्लास ग्रेड। त्यसमा चाहिँ करिब चाहिँ 23%मा चाहिँ कुनै न कुनै प्रकारको चाहिँ इम्पेयरमेन्ट पायौँ। अझ चाहिँ प्री र पोस्ट चाहिँ हामीले चाहिँ कम्पेयर गरेको छौँ। यो चाहिँ यो नम्बरमा चाहिँ यता तलमाथि छ, तर मतलब चाहिँ पर्सेन्टेजमा। तर यो चाहिँ सिग्निफिकेन्ट चाहिँ यो फरक चाहिँ सिग्निफिकेन्ट पाएनौँ हामीले। जुन प्याटर्नमा चाहिँ प्री इलिमिनेसनमा आउने गरेको बिरामी जस्तो प्याटर्न छ, पोस्ट इलिमिनेसनमा पनि त्यस्तै नै रह्यो। अनि यो हामीले चाहिँ त्यो इतिहास केलायौँ, कतै न कतै हाउस होल्ड कन्ट्याक्ट छ कि छैन भनेर। अनि चाहिँ करिब चाहिँ 11% चाहिँ यो स्टडी पपुलेसनमा चाहिँ हाउस होल्ड कन्ट्याक्ट चाहिँ पायौँ। त्यसमध्ये चाहिँ प्यारेन्ट धेरै 7 छ, अनि सिस्टर भनेर चाहिँ 4 छ, बाँकी चाहिँ अरू रिलेसनमा उनीहरू कन्ट्याक्टमा रहेछ। अर्को चाहिँ हामीले चाहिँ यो ड्युरिङ अथवा चाहिँ एट द डायग्नोसिस टाइममा रियाक्सन छ कि छैन भनेर हेऱ्यौँ। करिब करिब 26.7%मा रियाक्सन डेभलप भयो। यो चाहिँ न्युराइटिस पनि हुनसक्छ, टाइप 1 or टाइप 2, तर मोस्ट अफ देम आर टाइप 1 रियाक्सन। अनि अर्को एउटा इन्ट्रेस्टिङ चाहिँ यहाँ तल छ, जुन चाहिँ 3.4% लेप्रोसी पेसेन्ट ह्याड ड्याप्सन हाइपर सेन्सिटिभिटी सिन्ड्रोम देखियो। यही केस हो, यदि राम्रोसँग क्योर गरेन भने मोर्टालिटीमा जाने है। अनि हामीले कन्क्लुजन ड्र गऱ्यौँ। लेप्रोसिस केसेस आर सिन इन डिक्रिजिङ ट्रेन्ड इन द पोस्ट इलिमिनेसन फेजमा इरामा। हाउएभर हायर proportion of MB cases during post elimination could be due to delay in diagnosis. Child cases an indicate of active transmission. हामीले चाहिँ चाइल्ड केस पनि पाएको छौँ, त्यो हो कि भनेर। अनि द एक्टिभ ट्रान्समिसन इन द कम्युनिटी लेभल। अनि रिजल्ट अफ दिस स्टडी अग टु निड यो स्ट्रेटेजी चाहियो अर्ली केस डायग्नोसिस, एक्टिभ केस फाइन्डिङको लागि, अनि रियाक्सन म्यानेज र डिसेबिलिटी प्रिभेन्ट अथवा म्यानेजको लागि चाहिँ यस्तो रणनीतिहरू चाहिन्छ भनेर ड्र गऱ्यौँ। हुन्छ, हामी एक्नोलेजमेन्टमा हामीले लेप्रोसी एफेक्टेड पिपललाई ल्यायौँ अनि लेप्रोसी मिसन नेपाललाई ल्यायौँ। हस्, थ्याङ्क यू भेरी मच।
Thank you for the wonderful presentation. We would like to take 2 questions from the floor.
हजुर।
एकदमै समय सापेक्ष जस्तो लाग्यो तपाईँको प्रेजेन्टेसन, द वे यु डिलिभर्ड वाज अल्सो गुड। तर मैले एउटा प्रश्न, तपाईँले सुरुमा एक्टिभ केस फाइन्डिङको जुन कुरा हाइलाइट गर्नुभयो नि, तपाईँले यति नम्बर थियो यति पछि थियो, बट एक्टिभ केस फाइन्डिङको लागि जुन केही निचोड आउला जस्तो लागेको थियो त्यो चाहिँ देखिएन।
हामीले चाहिँ अहिले ललितपुर जिल्लामा चाहिँ खालि चाहिँ क्याम्पहरू गऱ्यौँ 2-4वटा, अनि प्लस चाहिँ यो कन्ट्याक्ट ट्रेसिङ गऱ्यौँ। त्यतिले मात्रै पुग्दैन भन्ने यो इन्डिकेसन हो कि अब अरू केही गर्नुपर्छ कि अरू नयाँ मोडलमा, होइन? त्यो पुरानो मोडल हो कि यो।
I was thinking that and the take home message would be okay, यो प्रकारको केस फाइन्डिङ गर्दा एक्टिभ केस फाइन्डिङ गर्दा बेटर होला भनेर।
अझै हामी चाहिँ जिरोमा ल्याउने हो भने यसले मात्रै पुग्दैन भन्ने छ।
थ्याङ्क यू।
यता आउनुहोस्।
थ्याङ्क यू सर एगेन। मैले नेपालीमा भन्छु सबैजना बुझ्नु। मलाई 2टा कुरा चाहिँ यसबाट मैले बुझेको छु। एउटा कुरा त 2010 मा इलिमिनेसन हुँदाखेरि एउटा क्वेसन उठे थियो कि यो कहीँ न कहीँ गभर्मेन्टले इलिमिनेसन अलि पहिले त्यतिकै घोषणा गरेछ भनेर, तर इलिमिनेसन सही थियो भनेर त्यो यो पुष्टि हुन्छ। 2010 को इलिमिनेसन पछि ललितपुर जिल्ला एउटा उदाहरण हो जो सस्टेनेबली तल गइरहेछ डिजिज भनेर एचिभमेन्ट एउटा मैले त्यो हेरेँ। त्यसको लागि चाहिँ यो स्टडीले त्यसलाई पुष्टि गर्छ। दोस्रो कुरा, कम्युनिटीमा अहिले पनि चाहिँ केस छ। एउटा चाइल्ड हेल्थ चाइल्डको कुरा हेऱ्यो त्यो 5% जति छ, हैन? ग्रेड 2 डिसेबिलिटी, यो 2टा इन्डिकेटरले कहीँ न कहीँ ट्रान्समिसन देखाइरहेछ। अब चाहिँ म्याडमले कुरा गर्नुभएको थियो कहाँ हिट गर्ने भनेर। अब मोडेल चाहिँ हामीले नि दिने हो पोलिसी लेभललाई। अब कुन मोडेलमा जति मोडेल छ एक्टिभ केस डिटेक्सनको त्यसबाट कमै केस आएछ कि पहिले प्यासिभ केस डिटेक्सनबाट नै म्याक्सिमम केस आउँछ। अब न्यू मोडल न्यू जेनेरेसनले सजेसन गर्नुहोस् कसरी बाई रिसर्च र पोलिसी मेकरलाई सजेस्ट गरेर कसरी चाहिँ इलिमिनेसनलाई सस्टेन गर्ने। जुन अहिले तराईको करिब 10वटा जिल्ला 12वटा जिल्लामा अहिले इलिमिनेसन चाहिँ एचिभ गर्नुपर्ने छ। त्यसको लागि पहिले फर्स्ट चरणमा हेरौँ। सेकेन्ड चरणमा मेरो सजेसन छ कि न्यू आइडिया लिएर आउँ र यसलाई चाहिँ अब इरिगेसनतिर जाउँ कि थ्याङ्क यू
Thank you, Dr. Thank you.
Thank you very much. Thank you, Mr. Ashish Shrestha, for the wonderful presentation. Now we'd like to move on to the fifth presentation. Our fifth speaker is Mr. Nishan Kotwal. Mr. Kotwal is currently working as in charge of molecular and genomic sequencing research lab and project manager at Center of Infectious Disease and Research and Surveillance Research Development Division, Tulikel Hospital. He has received MS in biotechnology from Norwegian University of Science and Technology, Norway, specializing in nanomedicine and pharmaceutical rheology. Thank you.
Thank you, sir. Thank you for this wonderful introduction. So I'm Nishan. I'm representing here our collaborative study that we did with International Vaccine Institute back in 2022. I'm presenting on seroprevalence, geospatial distribution, and risk factors of hepatitis C infection in Nepal. Okay, thank you. Yeah, great. So this is a group of investigators, a really collaborative group, a bunch of people from IVI, from Johns Hopkins, and the Tulikel Hospital. So a little bit of background. Everybody is well known with hepatitis E infection, which is known to cause acute jaundice syndrome, and often considered neglected disease, with the majority of infections due to poor surveillance as well as access to healthcare and access to good diagnostic as well. And most of the genotypes that are circulating is either one or two, mostly with fecal-oral route. And the global burden has, there are various studies, so the global burden is very variable, and it's estimated that 1 out of 8 people are actually have recent infection. So Nepal has had like 5 different HIV outbreaks until now, and it has been called an endemic in Nepal, which started in 1982, the outbreak, but there was one study back in 1973, which was published in Journal of Nepal Medical Issues, and I was very surprised to find that paper as well. So for the first outbreak outside the Kathmandu was in Biratnagar back in 2014, and this was a very interesting study, which affected nearly 7,000 people, and they were actually also were able to identify the genotype as well by using partial 412 nucleotide reason. But apart, one of the reason of doing this study is Nepal actually doesn't have much of representative data on hepatitis C. That is actually national representative. Sorry, I keep on using this. So this shows a global distribution of hepatitis E all over the world, and was like the slide on the right actually is another study that we did in 2021 and 22, where we did just in in Kathmandu Valley and in Tulikel, which was a longitudinal study, but still I wanted to highlight that there is not much nationally representative data. So this shows the general transmission of hepatitis E through fecal-oral route with our food from people with donated contaminated bloods and so on. So the objectives of our study was to first to estimate the hepatitis E seroprevalence, which is representative. The other one, to see the geospatial distribution of hepatitis E in all over Nepal, and also different factors associated with it. So the methodology that we used was one from a cholera study that was done by a team with IVI and Nepal Medical College, where they did the serosurvey for cholera study, and we used that same samples and few of those questionnaires to evaluate. I'll come as a take-home message for this one as well, and we took a different approval to use that sample to evaluate hepatitis E. 65 different wards all over Nepal, 15 households per ward, and people from 2 years above were evaluated. And we did ELISA, IgG ELISA to evaluate the samples. And we used differentiated them into urban and rural municipalities, and then delineated them into wards. And also, there are different various sources of water that are used all over Nepal. So we differentiated them into improved and non-improved as for WHO classification. And then we used the statistical seroprevalence to identify the seroprevalence as well, and use a mixed effect logistic regression model for the factors associated, as well as univariate and multivariate analysis. In univariate, we used individual level and ward level, where individual level, age, gender, water drinking habits, and multivariate level, we did a full model as well as a reduced model. I presented a full model here, but in our publication from later on, please follow that if you have time. We have used reduced model as well, just focusing on the factors that are very closely associated with hepatitis E virus seropositivity. For example, a person with older age male and living in a hilly region. And then we also did a spatial distribution using hierarchical logistic spatial regression model. This is a very good model. It has some downside as well. But the one good thing, we used a 5 by 5 kilometer radius and used like national published data and as well as publicly available data for, travel time, for elevation. But one of the downside is actually it assumes that there's a smooth change in hepatitis C seroprevalence, which is not always true because there are other factors like environmental factors, behavioral factors, healthcare access, availability, which could differ, and that has not been incubated into the spatial regression model. So coming to the results, we can actually see that we didn't see much difference in urban and rural areas in the seroprevalence, which is a bit different from other study which actually has some brought into urban areas as well, because of population density. But what we did find is the hilly region actually had higher seroprevalence than the Tarai and mountain region. Could be due to population density and also the quality of water, unmonitored water. Regarding the age, what we have actually seen, does this point as well? Okay, never mind. What we also saw is with age, the HIV seroprevalence increases because you get exposure over time. But there's one thing interesting what we have seen is after 19 years, there's a very steep increase in hepatitis C seroprevalence, and which goes on. And in all of this age group and in generalized as well, male population actually had higher prevalence of IgG compared to female population. Moving on, you can see the same thing. Like until 15 to 19 age group, there's just 4.3 of seroprevalence, which increases directly into 21% when you go to higher age. And this actually is same for most of these studies that has been done worldwide. Like with age, the seroprevalence, seropositivity, it increases. With highest being the highest age group here. And same with the male and lower income people. With the drinking water, also unimproved drinking water, which are like open sources. And unmonitored sources, they have high risk of HIV infection. Same with the unimproved toilet facility, and Bagmati province having the highest out of all of the provinces, with Kathmandu having as high as 50%. And then, as I said, mentioned before, like hilly region having higher HIV seroprevalence compared to Tarai and mountain regions. So this is where we did the spatial distribution all over Nepal, where we usually 5 by 5 kilometers grid cells to make them. And all of the gray dots are actually the sample locations from hilly, mountain, and Tarai region. And the big star that you see is our Kathmandu, which has the highest seroprevalence, which is as high as 50%. The lowest was in the Tarai region, which was as low as 10%. So concluding, the overall seroprevalence was 20.8. There is no significant difference between urban and rural areas, as I mentioned before, but it's contrastingly higher in hilly and mountain regions, which could be due to lack of access to clean water, as well as unmonitored water and urbanized and population density as well. The highest risk of infection was in Kathmandu Valley, which is not too surprising. But what we did see is very steep rise in seropositivity after 20 years, because there might be some eating habits, going out, working population, so that might actually increase or add on to the HIV infection. One of the take-home messages for this is we can use IgG as a marker of infection, as a proxy marker. But there's a little bit downside to it as well, because IgG actually wanes over time. So there are some studies that actually showed that, after I think around 6.5 years or something, around 7 years, the IgG antibody actually decreases by 23%. So we have to be very careful. We might be underestimating the burden sometimes when we are using IgG as a marker. The other thing is the residual sample investigation that I mentioned about. We use the samples for this study that was initially called a part of a cholera study. We did a different ethical approval, we approved it, and we did this study. So it actually, saves a lot of resources, a lot of logistically and funds as well. So we are using the same samples and just using a different laboratory evaluation to that, which it could be, to do several studies, related studies of public health concern, for example, with COVID as well. So the other take-home message is, good water, health, sanitation, and hygiene interventions along with HIV vaccine. There's just one vaccine as of now. I think it's Helicon or something, which has been approved and just being used in China and in Pakistan for now, which can be used. And actually WHO recommends to use it for high-risk population as well as pregnant women as well. So maybe we have to think about there as well. I think they did a phase 3 study which had an efficacy of 100% or something, as far as I remember. And what is the other thing also is the thing that I mentioned earlier about IgG, gradually decreasing. There's, there's instances of seroreversion in some of the, and mostly in children. That's why that particular population should also be studied. But again, we have to be very careful that we should not be underestimating the risk of infection by using IgG. Because the other thing is what some studies have found is the IgG actually wanes over time very quickly when there's a natural infection when compared to the vaccine. So that adds to another, reason of using the vaccines as well. So lastly, I want to acknowledge NHRC for this wonderful summit, which is in their 10th version now, and also Nepal Medical College and New Era, which were part of the initial cholera study. Huge thank you if any of them are here from that institution, and to IVI for funding this study, and Dhulikhel Hospital and Kathmandu University Medical School of Sciences. Thank you so much. Thank you for your time. Any questions? Yeah, thank you.
Thank you very much. The floor is open for the question.
Thank you, Nishan sir, for a very nice presentation. So, one thing that struck me while you giving your presentation is that, please would you put few words on male and female prevalence? It means that you said that there is a variation. Yeah, male have a high prevalence than female. So, please few words.
Okay, so this is something we have found similar in our other longitudinal study as well. Maybe traditionally, the male, they tend to go out more in the rural population to work and eat out more. So that could be one of the reasons they have a little bit of higher hepatitis C seroprevalence compared to the female. Yeah, couldn't be very relevant to current urban aspect. But when when you do a multivariate analysis and put everything together, that could be one of the reasons that the male population has a higher IgG prevalence than the female has, and that's with time. So. Yeah.
सो कन्सर्न चाहिँ के भन्दाखेरि अहिले भर्खरै पनि तपाईँले भन्नुभयो कि मेल र फिमेलमा चाहिँ भेरिएसन छ अनि फेरि कन्क्लुजनमा चाहिँ आइजिजी क्यान बी युज एज अ मार्कर भन्नुभयो होइन फेरि तपाईँले के पनि भन्नुभयो त्यो इट्स वेन्स विथ टाइम पनि भन्नुभयो त्यसो यो दुईवटा अवस्थामा हामीले कसरी त्यसलाई चाहिँ मार्करको रूपमा युज गर्न सक्छौँ त पहिलो कुरा त आइजिजी आफैमा पनि अलिक टाइम लाग्छ त्यसलाई एक्टिभेट हुनको लागि होइन भेरिएसन छ मेल र फिमेलमा सो वाट्स द थ्रेसहोल्ड टु बी कन्सिडर्ड फर बिङ a marker for HIV?
Oh, thank you for the question. So I think it relates to one of our other studies, so I'm just plugging another study that we did. So maybe longitudinal study could be one of those idea where we follow the same patient and also see how IgG is decreasing over time. So incorporating that into the study could be one of the ways you can use IgG as a proxy marker. So it's just a proxy marker to use and see the prevalence of an infection. I'm not, technically an epidemiologist, so maybe some of the epidemiologists over here could add on to that. But I think doing a longitudinal study and following them over time and seeing, how that IgG actually wanes over time and what's the time that it actually does, maybe a few years, few months. So incorporating that in this study, we can use it as a proxy marker. Yeah, thank you.
Thank you very much. Now we would like to move on to...
Yeah, thank you so much. Thank you for your time.
The sixth presenter. Our sixth presenter is Dr. Pooja Shrestha. Dr. Pooja is a project manager for ACT South Asia study, the azithromycin and cefixime treatment of typhoid in South Asia trial at Okru, Nepal. Her responsibilities include managing the necessary actions for trial, clinical evaluation of patient enrolled in the trial, communicating with the project team within Nepal and project site countries, and providing training regarding the trial protocol. Over to you.
So, namaste everyone. Thank you, respected chair and co-chair. I would also like to thank NHRC team for providing me the opportunity to present my presentation in today's platform. So I would like to use both the language during my presentation. So my study topic is azithromycin and cefixime combination versus azithromycin alone for the outpatient treatment of clinically suspected or confirmed uncomplicated typhoid fever in South Asia. It's a randomized controlled trial. This study is ongoing study. In short, we call our study as ACT South Asia. So the country participating in this country is Nepal, Bangladesh, and Pakistan. Nepal is the leading country for this study. So I call this study as multi-centered and multi-country because in Nepal we have 6 sites, so namely Patan Hospital, Civil Service Hospital, Siddhi Memorial Hospital, BPKIHS Dharan, Sukuras Tropical Infectious Disease Hospital, and Shir Memorial Hospital. And from Pakistan we have Aga Khan University Hospital, and from Bangladesh we have International Center for Diarrheal Disease Resource Center. So we are affiliated with Oxford University Clinical Research Unit, Nepal and Vietnam, Aga Khan University, ICDDRP, Liverpool School of Tropical Medicine, and our study main PI is Dr. Buddha Basnet. So in the background, I would like to focus you all regarding the typhoid cases. So we know typhoid cases are very much common in our community, especially in the low and middle income country. So we have incident rate of around 500 per 100,000 population in the communities of South Asia. So usually in the treatment of typhoid, our physician use azithromycin as the treatment of choice. We might know that azithromycin the typhoid organism, typhoid-causing organism, Salmonella species, are mostly in the intracellular level, but there are still the bugs present in the extracellular level as well. So from the various research, we can see that azithromycin works in the intracellular level, and cefixime works in the extracellular level. So what we hypothesize is a combination of both the drug would be better in preventing treatment failure in suspected typhoid cases and uncomplicated fever cases. So we move ahead with the objective. So our main objective is to determine whether treatment with 7 days of an azithromycin and cefixime combination is superior to 7 days of azithromycin and placebo in preventing treatment failure in patients with clinically suspected or confirmed uncomplicated typhoid fever. So यसमा चाहिँ मुख्य हामीले के हेर्न खोजेको छौं भन्दाखेरि चाहिँ क्लिनिकल फेलियर र माइक्रोबायोलोजिकल फेलियरमा चाहिँ हामीले यो आउटकम चाहिँ डिसाइड गरेको छौं। जस्तो क्लिनिकल फेलियरमा चाहिँ हामीले के हेर्न खोजेछौं भने डे 7 सम्म चाहिँ अब बिरामीलाई हामीले ट्रिटमेन्ट दिन्छौं। सो डे 7 मा उसको फिभर कस्तो छ त, उसको फिभर सबसाइड भयो कि भएन हैन, यदि त्यसमा फिभर सबसाइड भएन या कुनै कम्प्लिकेसन आयो भने चाहिँ यो हामी क्लिनिकल फेलियरमा राख्न सकिन्छ। र त्यसै गरेर 28 डेज भित्र फेरि पनि फिभर रिल्याप्स भयो अनि कुनै कम्प्लिकेसन्सहरु आयो भने पनि हामी यसलाई चाहिँ क्लिनिकल फेलियरमा क्याटेगोराइज गर्न सक्छौं। र त्यसै गरी माइक्रोबायोलोजिकल फेलियरमा पनि हामीले चाहिँ डे 7 या 28 डेज भित्र फेरि पनि कल्चर पोजिटिभ आयो या अरु कुनै कम्प्लिकेसन ग्रोथ भयो भने यसलाई चाहिँ हामी माइक्रोबायोलोजिकल फेलियरमा राख्ने छौं। सो मेथोडोलोजीको कुरा गर्नु पर्दाखेरि चाहिँ हाम्रो यो ongoing study हो। सो यो स्टडी चाहिँ it is a comparative participant and observer 1:1 double blind randomized control trial phase 4 study हो। सो यसमा चाहिँ म प्रोसेस भन्नु अगाडि चाहिँ छोटो हामीले चाहिँ हामीले चाहिँ पेशेन्टलाई चाहिँ 1:1 ग्रुपमा चाहिँ र्यान्डमाइज गरेको हुन्छ र सो यसमा चाहिँ आर्म ए मा चाहिँ हामीले एजिथ्रोमाइसिन 20 mg per kg per day oral dose once day and cefixime 20 mg per kg per day oral dose in 2 divided dose for 7 days दिन्छौँ भने आर्म बी मा चाहिँ हामीले एजिथ्रोमाइसिन 20 mg per kg per day oral dose once daily for 7 days अनि cefixime matched placebo पनि दिन्छौँ यो ग्रुपलाई चाहिँ। सो यसमा चाहिँ प्लेसिबो भन्नु बित्तिकै चाहिँ कन्फ्युज नहुनु होला बिकज दुईटै ग्रुपलाई चाहिँ हामीले एजिथ्रोमाइसिन चाहिँ दिएको हुन्छ। सो सेफिक्सिन मात्रै हाम्रो र्यान्डमाइज भएको हुन्छ। सो हाम्रो मेथोडोलोजीको कुरा गर्नु पर्दाखेरि चाहिँ हाम्रो टार्गेट स्याम्पल साइज चाहिँ 1500 रहेको छ सो यसमा चाहिँ हामीले कसरी प्रोसेस गर्छौँ कसरी हाम्रो ट्रायल स्किममा कस्तो हो भने चाहिँ हाम्रो CMAsहरू चाहिँ डिफ्रेन्ट OPDsहरूमा हामीले चाहिँ पोस्ट गरेको हुन्छ र उहाँहरूले चाहिँ हाम्रो इन्क्लुजन क्राइटेरिया जस्तो फिभर 37.5 डिग्री अबोभ हुनुपर्ने अनि 42 आवर्स भन्दा बेसी ज्वरो आएको बिरामी र कुनै पनि एन्टिबायोटिक नलिसकेको बिरामीहरूले चाहिँ स्क्रिन गरेर हाम्रो OPDमा ल्याउनुहुन्छ र त्यसै गरी हाम्रो OPDमा चाहिँ मेडिकल अफिसर्सहरूले चाहिँ उहाँको विभिन्न इन्क्लुजन र एक्सक्लुजन क्राइटेरियाहरू स्क्रिन गरेर उहाँहरूलाई चाहिँ क्लिनिकली र फिजिकली एक्जामिन गरिसकेपछि चाहिँ उहाँहरूको ओरल कन्सेन्ट लिएर चाहिँ हामी RDT पर्फर्म गर्छौँ सो RDT भन्नाले ऱ्यापिड डायग्नोस्टिक टेस्ट चाहिँ हामी डेङ्गी मलेरिया स्क्रब टाइफस र CRPमा गर्छौँ सो हाम्रो स्टडीको एन्ट्री क्राइटेरिया चाहिँ CRP लेस देन 10 हुनुपर्छ र त्यसै गरी डेङ्गी स्क्रब टाइफस र मलेरिया चाहिँ नेगेटिभ हुनुपर्छ यदि यो सबै अब कुनै मध्ये कुनै RDT पोजिटिभ आयो भने चाहिँ हामीले उहाँ पेसेन्टलाई चाहिँ जेनरल गाइडलाइन अनुसार ट्रिटमेन्ट गर्छौँ र केही गरी उहाँको सबै RDT नेगेटिभ छ CRP पनि मोर देन 10 छ र द पेसेन्ट इज विलिङ टु पार्टिसिपेट हामीले चाहिँ उहाँलाई फर्दर काउन्सिलिङ गरेर रिटर्न कन्सेन्ट लिन्छौँ र अघि डिभाइड गरेको दुईवटा मेडिसिन को ऱ्यान्डमाइज नम्बर चाहिँ पिक गरेर उहाँलाई चाहिँ त्यो इन्भेलपको मेडिसिन चाहिँ उहाँको बडी वेट अनुसार चाहिँ उहाँलाई प्रिस्क्राइब गर्छौँ र हामीले चाहिँ उहाँलाई सबै औषधि खाने तरिका औषधिको साइड इफेक्ट्सहरू सबै नै हामीले एक्सप्लेन गरेका हुन्छौँ र त्यसमै अब फलोअप प्रोसेस पनि सुरु हुन्छ जस्तो उहाँ पेसेन्ट इनरोल हुनुभएको दिनदेखि डे 7 सम्म हाम्रो रिसर्च नर्सले चाहिँ बिहान बेलुकै फोन गरेर उहाँहरूको चाहिँ अब फिभर कति छ अरू केही कम्प्लिकेसन या केही इभेन्ट्सहरू भएको छ भनेर चाहिँ हाम्रो डेटामा नोट गर्नुहुन्छ त्यसै गरेर डे 2 र डे 4 मा चाहिँ हाम्रो कम्युनिटी वर्कर्सहरू चाहिँ पेसेन्टको घरमै जानु भएर उहाँहरूको चाहिँ भाइटल्सहरू लिएर उहाँहरूले औषधि खानुभएको तरिका मिलेको छ कि छैन कुनै अरू कम्प्लिकेसन्सहरू छ कि छैन भनेर चाहिँ हाम्रो क्लाइरिस डेटामा रिपोर्ट गर्नुहुन्छ र फलोअपको कुराहरू चाहिँ डे 7 डे 14 र 28 मा चाहिँ हाम्रो हस्पिटलमै फलोअपको लागि बोलाउँछौँ र कल्चर पोजिटिभ डे 0 मा हुनुहुन्छ भने चाहिँ हामी 90 डेजसम्म फलो गर्छौँ सो मैले पहिल्यै भने हाम्रो चाहिँ यो अनगोइङ स्टडी हो सो हामीले चाहिँ यो स्टडी लगभग कोभिडको एकदम हाई सर्जको टाइममा स्टार्ट गर्यो समवेयर अराउन्ड मे 2021 र मलाई अझ पनि याद छ त्यो स्टार्टिङ फेजको पहिलो 5-6 महिना चाहिँ हामीले 8-9 जना बाहेक हामीले एनरोलै गर्न सकिरहेको थिएनौँ तर बिस्तारै हामीले साइट एडिसन्सहरू गऱ्यौँ र अहिले यो विदिन दिस 2 years वी हेभ बिन एनरोलिङ मोर देन 1200 पेसेन्ट फर दिस स्टडी अनि तीमध्ये लगभग 20% को चाहिँ ब्लड कल्चर पोजिटिभिटी पनि पाएका छौँ र त्यसै गरेर हामीले चाहिँ स्टडीको हाफ अफ द पपुलेसन अब 1500 मा 750 पपुलेसन लिएर चाहिँ हामीले एन्ट्री मेनलाइसिस पनि हामीले पर्फर्म गर्यौँ सो एन्ट्री मेनलाइसिस चाहिँ हाम्रो डिसिजन मेकर्सहरू हुनुहुन्छ स्टियरिङ कमिटी र DSMB डेटा सेफ्टी मोनिटरिङ बोर्ड सो उहाँहरूलाई चाहिँ हामीले डेटा दिएपछि उहाँहरूले चाहिँ साइलेन्टली पर्फर्म गर्नुहुन्छ र यसको रिजल्ट चाहिँ हामीलाई भनिसक्नुहुन्न सो उहाँहरूले नै पर्फर्म गरेर त अब यो स्टडी चाहिँ आउटकम राम्रै आउँदैछ सो यु क्यान कन्टिन्यू भनेर अप्रुभल पाइसकेपछि चाहिँ हामीले यो स्टडी अझ पनि कन्टिन्यू नै गरिरहेका छौँ र होपफुली यो चाहिँ हामीले नेक्स्ट इयरसम्म कन्टिन्यू गर्छौँ अब डिस्कसनको पाटोमा चाहिँ हाम्रो यो स्टडी अनगोइङ हो अब रिजल्ट त हामीसँग छैन अब हामीलाई चाहिँ के रिजल्ट आउँछ भन्ने चाहिँ वी डोन्ट नो सो वी आर इन द स्टेज अफ इक्विपोइज होइन जसमा चाहिँ एउटा अनसर्टिनिटी छ जस्तो के हुन्छ त रिजल्ट होइन अब रिजल्ट चाहिँ हाम्रो हाइपोथेसिसको फेभरमा पनि आउन सक्छ अथवा यसको उल्टो पनि हुनसक्छ दुईवटा कम्बिनेसनले पनि राम्रो ट्रिटमेन्ट दिएको हुनसक्छ या एजिथ्रोमाइसिन अलोनले मात्र पनि वर्क गरेको हुनसक्छ सो अब हामी सबैजना अस्तिदेखि नै एभिडेन्स बेस्ड साइन्टिफिक क्लिनिकल ट्रिटमेन्टको कुराहरू गर्दैछौँ सो यस वी आर हामीले चाहिँ रिसर्च गरिरहेछौँ हामी एभिडेन्स खोजिरहेछौँ होइन अब यही क्रममा चाहिँ अब हाम्रो जस्तो रिजल्ट आउँछ हामीले चाहिँ यो रिजल्टलाई चाहिँ हामीले अगाडि बढाउँछौँ र रिजल्ट आउटकमहरू सबै आइसकेपछि हामीले एउटा रिनाउन्ड पियर रिभ्यू जर्नलमा पनि पब्लिकेसन गर्छौँ र त्यसपछि हामी आउनेछौँ पोलिसी मेकर्स डिसिजन मेकर्सकोमा उहाँहरूलाई रेकमेन्ड गरेर चाहिँ यो अब क्लिनिकल प्र्याक्टिसमा इम्प्लिमेन्ट गराउन रिकमेन्ड गर्छौँ र त्यसै गरेर अब हाम्रो चाहिँ क्लिनिकल प्र्याक्टिसनर्सहरूलाई पनि हामीले चाहिँ अब यस्तो रिजल्ट आएको छ भन्ने हिसाबले चाहिँ हामीले यो प्र्याक्टिस अगाडि बढाउन चाहिँ उहाँहरूलाई पनि रेकमेन्ड गर्छौँ सो मेरो भनाइअनुसार चाहिँ अब हामी रिसर्च गर्छौँ तर रिसर्च गरेपछि यो इम्प्लिमेन्टेसन भइरहेको हुँदैन कतिपय ठाउँमा सो हामीले चाहिँ फाइन्डिङ्सहरू चाहिँ सबैजनाले चाहिँ एउटा राम्रोसँग बुझेर चाहिँ यसलाई चाहिँ इम्प्लिमेन्ट गर्न चाहिँ अगाडि बढाउँ भन्न चाहन्छु सो We thank our partner, Okro Nepal, Patan Hospital, Aga Khan University, Oxford University, International Center for Diarrheal Disease Research Center, Liverpool School of Tropical Medicines, अनि हाम्रो फन्डर्स हुनुहुन्छ युनिभर्सिटी अफ अक्सफोर्ड अनि हाम्रो स्पोन्सर हुनुहुन्छ युनिभर्सिटी अफ अक्सफोर्ड अनि हाम्रो फन्डर्स द जोइन्ट ग्लोबल हेल्थ फन्ड एमआरसी डेफिड र वेलकम ट्रस्ट सो दिज आर आवर रेफरेन्स सो वी थ्याङ्क यू
Thank you, Dr. Pooja. Floor is now open for the question.
Namaste. I'm Renu Karke. I'm a pharmacist. And my concern is that I have a query. You patients are self-medication पनि गरे हुन सक्छ नि त हैन सो त्यसको as a confounding variable तपाईको स्टडीमा चाहिँ के इम्प्याक्ट हुन सक्छ किनभने पेशेन्ट चाहिँ घरमा गइसक्छ हैन त्यसपछि चाहिँ त्यो फिभरको पेशेन्टले अरु कुनै मेडिकेसन सेल्फ मेडिकेसन गरेको कन्टेक्स्ट ओभर द काउन्टर पनि मेडिकेसन लिन सक्छ नि त हजुर सो तपाईले भन्नलाई चाहिँ फोन गरेर सोध्छ नर्सहरुले चाहिँ भन्नुभयो हैन तर यसको अरु कुनै कन्फाउन्डिङ भेरिएबल्सहरु त्यसको चाहिँ म्यानेजमेन्ट कसरी गर्छ यो ट्रायलले
Thank you for the question. So, सुरुमा हामीले स्क्रिन गर्दाखेरि नै पेशेन्टहरुलाई चाहिँ उहाँहरुको मेडिसिन हिस्ट्रीको बारे सोधेको हुन्छौँ हैन अब उहाँहरु कति पहिले त थाहा हुँदैन अलि के लिएको भनेर त्यसपछि हामीले चाहिँ उहाँलाई त्यो दिन घरमै पठाएर चाहिँ के औषधिहरु खानु भएको छ मिल्छ भने भोलि आउनु भनेर पनि पठाउँछौँ नभए चाहिँ मेसेन्जरबाट या फोटो खिचेर भाइबरमा हामीले औषधिहरु पठाउनु पनि भन्छौँ अब हाम्रो चाहिँ हाम्रो एन्ट्री क्राइटेरिया भनेको खाली हाम्रो स्टडी मेडिसिन एजिथ्रोमाइसिन र सेफिक्सिन मात्रै नलिनु हो हैन त्यो बाहेक अन्य मेडिसिनहरु लिइरहेछ भने वी आर the patient is allowed for the enrollment तर त्यसपछि पनि अब उहाँले अब हाम्रो स्टडीको मेडिसिन खाइराख्नु भएको छ र त्यसबाफत फेरि अरु पनि औषधिहरु खाइराख्नु भएको छ भने चाहिँ हाम्रो नर्सेसहरुले चाहिँ डेली फोन गर्नुहुन्छ या होम भिजिट जानुहुन्छ सीएमएहरु सो उहाँहरुले चाहिँ राम्रोसँग काउन्सिलिङ गरेर चाहिँ सबै हेर्नुहुन्छ
म वशिष्ठ रिजाल म प्रोफेसनली माइक्रोबायोलोजिस्ट हो डाक्टर पूजालाई धेरै बधाई भन्न चाहन्छु यो मल्टी सेन्टर मल्टी कन्ट्री स्टडीको लागि तर एउटा दुईटा यो त अब धेरै एउटा विद्वानहरूले नै अवश्य गरे होला यो क्लिनिकल ट्रायल यो साल्मोनेला टाइफाइड यो चाहिँ नेपालमा त अल सिजन अल मन्थ भने 12 महिना आइसोलेट हुन्छ यो यसमा पनि यो विशेष गरेर हाम्रो रेनी सेसन सुरु भएपछि चाहिँ एकदम इन्सिडेन्ट्स बढ्छ यो पब्लिस गरेका छौँ हामीले अरू धेरैले पनि पब्लिस गर्नुभएको छ होइन अब यो केस नपाउने कुरा होइन यो कन्फर्म केसहरू नि अवश्य पाइन्थ्यो होइन तर यो सस्पेक्टेड केसमा क्लिनिकली सस्पेक्टेड केसमा क्लिनिकल ट्रायल गर्न किन आवश्यक पर्यो मेरो क्वेसन तपाईहरुको रिजल्टमा मेरो कुनै पनि त्यस्तो खालको क्वेसन होइन कि जुन ठाउँमा चाहिँ एउटा आइसोलेट ब्याक्टेरिया नै आइसोलेट भइरहेको छ कन्ट्रीमा जस्तो नेपालमा हुन्छ भने अवश्य पाकिस्तानमा पनि भएको होला बङ्गलादेश पनि अवश्य हुन्छ नि त्यसमा चाहिँ क्लिनिकली जस्तो सस्पेक्टेड केस भन्नुभयो हुन त अब तपाईहरुले यु हेभ डन दिस स्टडी हेज डन मेनी एक्सक्लुसन जस्तो टाइफस हैन मलेरिया अरू डिजिजहरू चाहिँ रुल आउट गरेको छ र एक्युट इन्फ्लामेसनको लागि त्यो सीआरपी पनि गर्नुभएको छ तर क्लिनिकली केस ब्याक्टेरिया आइसोलेट नभएको ठाउँमा त गर्न एकदमै राम्रो थियो तर यो ब्याक्टेरिया आइसोलेट भइराख्ने अब कति नेपालमा त एकदम एनी टाइम साल्मोनेला टाइफाइड आइसोलेटेड भइरहेकै छ भनेपछि यस्तो के कुरा हो डाक्टर साहबलाई केही थाहा छ कि भनेर मैले सोध्न खोजेँ मेरो क्वारी तर तपाईहरुको स्टडीमा मैले क्वेसन गरे हैन यो धन्यवाद
सो थ्याङ्क यू सो यसमा पनि अब हामीले हेर्न खोजेको चाहिँ मेनली कम्बिनेसन ड्रगले चाहिँ बेटर गर्छ कि गर्दैन rather than using a single drug हैन सो आइसोलेट हामीले अब ब्लड कल्चर कन्फर्म भइसकेपछि पनि अब कम्बिनेसन थेरापीले चाहिँ उहाँको फिभर क्लियरेन्स टाइम उहाँको कुनै अरू कम्प्लिकेसन्सहरू चाहिँ हेर्न खोजेको हो सर यसबाट सो त्यही नै भन्न चाहन्छु
नमस्कार मेरो सानो क्वेरी हो तपाईलाई धन्यवाद यो मल्टी सेन्टर स्टडी र मल्टी कन्ट्री स्टडीमा मैले खासमा जान्न खोजेको यो अघि सरकै लाइनमा के भन्दै डायग्नोज्ड केसमा एन्टिमाइक्रोबियल सेन्सिटिभ टेस्ट गरिकन दुईटा ड्रग दिने हो अथवा एउटा ड्रग दिने हो कि त्यतिकै ऱ्यान्डमली दिने हो?
यो चाहिँ हामीले ऱ्यान्डमली नै दिन्छौँ सर, ऱ्यान्डमाइज गरिएको हुन्छ। सो दुईटै ग्रुपलाई हामीले एजिथ्रो चाहिँ कमनै हुन्छ, तर एउटा ग्रुपलाई चाहिँ सेफेक्जिन पर्छ भने अर्को ग्रुपलाई चाहिँ प्लेसिबो भएको हुन्छ सर।
तर एजिथ्रो कुनै इन्डिभिजुअलको केसमा होइन, जब हामी कल्चर गरेरै रिपोर्टिङ गरिसकेका छौँ भने एन्टिमाइक्रोबियल सेन्सिटिभ गरेर दुईटै सेन्सिटिभ भएको नभएको केसमा दिँदाखेरि अलिकता उसको जुन एन्टिबायोटिकको लोड हुन्थ्यो त्यो अलिकता कम हुन्थ्यो कि त्यो नगरिकन दिने हो होइन सिधै?
सर हामीले ब्लड कल्चर गरेपछि चाहिँ
ब्लड कल्चर गर्दाखेरि तपाईँको रिपोर्ट आयो, टाइफाइड देखियो, सेन्सिटिभिटी नगरिकन ड्रग चलाउने कि सेन्सिटिभिटी गरिकन ड्रग चलाउने?
यो चाहिँ नगरिकनै हामीले पहिला चलाउँछौँ सर, तर यसमा कुनै रिजल्ट अब कुनैको रेसिस्टेन्ट आयो भने चाहिँ हामीले त्यो ड्रग स्टप पनि गर्छौँ सर। अनि योसँग हामीसँग रेस्क्यु ट्रिटमेन्ट पनि छ, हामीले यसको लागि चाहिँ सेफ्टी एक्सन युज गर्छौँ सर।
रेसिस्टेन्टको मतलब तपाईँले एन्टिबायोटिक रेसिस्टेन्टको लागि टेस्टिङ त पठाउने होला
सर? अलिकति क्लियर
होइन, मैले भनेको जुन एन्टिबायोटिक सेन्सिटिभ टेस्ट गर्नलाई
हजुर
पठाउने, रिपोर्ट नआउँदै तपाईँले मेडिसिन चलाउने, त्यसको रिपोर्ट आएपछि फेरि आफ्नो ड्रगको ट्रिटमेन्ट चेन्ज गर्ने, त्यो प्रोटोकल हो?
हजुर सर।
ओके,
thank you very much, Dr. Pooja.
Thank you,
we'll move
everyone.
on to the next presentation. Our seventh speaker is Professor Rajni Malla. She is professor at Central Department of Biotechnology, Jeevan University since February 2008. With over 16 years of experience in this role, the Central Department of Biotechnology benefits from their extensive expertise and leadership. As a prominent figure in the department, their contribution to teaching, research, academic administration and have been invaluable. Over to you, sir.
Respected Chairperson and namaste to all of you. Today I am here to present my research topics on application of lytic bacteriophage against carbapenem-resistant biofilms, resistant Klebsiella pneumoniae biofilms, and cloning of Holland gene responsible for bacterial lysis. My principal supervisor is Professor Dr. Rajni Malla. And regarding the antibiotics and bacteriophage, as you know that the carbapenem-resistant Klebsiella pneumoniae can cause the global and significant clinical threat because of lack of the therapeutic options, and so there is a worrisome about the about that, and because last resort of antibiotics like tigecycline and polymyxin and cholesterol sulfate developed the resistant, and as you know that the biofilm is one of the major virulence factor of MDR bacteria, and but antibiotics cannot degrade the biofilm producing bacteria, but in case of the bacteriophage, which can produce some types of fast proteins like choline, indolysine, and spannin, which have the capacity to degrade the biofilms, and so it is one of the alternative therapeutic options regarding MDR bacterial infections. The main objective of our research is application of lytic bacteriophage against carbapenem-resistant Klebsiella pneumoniae causing UTI, which can cause the UTI. And we have 3 work plans. Regarding work plan 1, we have isolated the MDR bacteria causing UTI, and which was confirmed by the 16S RNA genome sequencing, and their antibiogram was also performed, and resistant gene was also performed. And in work plan 2, we have isolated lytic bacteriophage, and their physiochemical properties was also characterized like a pH thermal stability and morphology also. And we also observed the effect of bacteriophage against biofilm tissue bacteria by using the scanning electron microscope. And in work plan 3, we have isolated and identified Holin gene responsible for bacterial lysis, and then Holin gene was also cloned and finally established a fast bank in CDBT. And here in this picture showed the antibiogram of a host bacteria by Kirby-Bott distribution methods, and bacteria was molecularly identified by 16S RNA genome sequencing, and resistant genes like NDM, whose base pair size was 621 base pair was identified. And this is the chromatogram of Klebsiella pneumoniae, and whose genome sequence was confirmed and or put into the NCBI blast, whose alignment approximately showed 97.2% similarity with Klebsiella pneumoniae. And this is the phylogenetic analysis of where this bacteria is closely related with the upper one bacteria. And here we can see that the phase isolation was performed by the double layer agar assay methods, and for the purifications purpose continuous streaking method was used, and also cesium chloride method was used, and ultracentrifuge also. And this picture clearly showed that the fast genome sequencing, where genome was extracted, and then finally library preparation was performed, and then that product was put into the Illumina MySeq, and the result was finally analyzed by using the bioinformatics tools. And here we can see that clearly we can see that the cloning and the protein expression of the phage protein, where phage genome was extracted, and then phage gene, that is the Holin gene, was amplified by using the PCR techniques, and that gene was ligated into the compatible vector, and that is also known as the recombinant DNA, recombinant vector, and that was transformed into a competent host, and then after that from that transformed cell we extracted the plasmid, and then transformed into the expression host, and then protein was extracted and purified by STS gel electrophoresis, and then quantification was performed by BCA technique, and after that protein expression will be performed in cell line culture or animal inoculations. Here we have isolated the phase that showed the bullseye appearance clearly and clear opacity, and was isolated from Kalimati rivers, and that was named 6697 phase. And this is the phase morphology which was observed under the transmission electron microscopy, and which belongs to the family Myoviridae family and Cordoviriales orders. And here we can see that the physiochemical properties of phage, where optimum temperature and pH, that is optimum temperature 37 and optimum pH 7 was where maximum plaque forming unit was obtained, and their latent period was 20 minutes was observed. And regarding the phage DNA, we have phage DNA was electrophoresis, and where their genome size was more than 10 kb. And then regarding the phage protein, we did the SDS page, where different types of protein was observed there of varying size. And here we can see that the circular view of whole genome sequence of Phage, where it contains the 45,288 base pairs, and certain proteins like indolysine, holin, and spanins proteins was also contained there, which is responsible for lysing the bacteria and as well as biofilm producing bacteria also. After that, we isolated and characterized the holin genes by using these primers, and where we can where the holin gene base pair was 252 base pairs, and their sequence was extracted by gel extraction kit method, and then put into the checked by the NCBI sequence, where their approximately size was 252 base pair we found. And after that we did the cloning, and for the transformed cells was confirmed by the blue-white screening method, where we can clearly see that the white colony showed the transformed colony, and from that white colony we extracted the plasmid and then run into the gel, where we can see that different bands was observed there. And after the transformation, we did the ligation, we did the restriction digestion by using restriction enzymes, single digestion and then double digestion also, and where we can see that the in L1 and L4, which contains the T vector plus holin, there are approximately size is about 4.2 kilo base pair, in the same way in double restriction digestion also L3. And in our plants, we have that cloned cell was sent for the sequencing purpose, and again that cloned cell was expressed in the PET28B vector, PET28 expression vectors for ligation, transformation, and restriction endonuclease enzyme. And finally, we did the biofilm production also by tissue culture plate methods, and after that we applied the Phage against the biofilm producing bacteria, and then sent to the AIIMS India for the scanning electron microscope, where we can see that this figure, that one shows that the numerous amounts of bacteria was on that site, but where biofilm treated with Phage showed the less number of bacteria, and also their cell wall was also going to degrade due to the application of the Phage. And now the conclusions: the Phage was found to be stable in different physiological conditions, and the take-home message is the Phage holin protein can be the next generation antibacterial agent to treat MDR pathogen if further research is carried out in clinical trial. And I acknowledge to my head of department, Professor Dr. Krishna Das Manandar, and the differences, and the mother of Phage in Nepal. Thank you.
Thank you for the presentation. The floor is open for the question.
धन्यवाद. म एकदम गजबको काम गर्नुभएको छ. मलाई पनि इन्ट्रेस्ट छ यो फाजमा, अलिअलि म पनि काम गर्छु. सो यसमा चाहिँ मेरो क्वेसन चाहिँ के भन्दाखेरि तपाईले जुन त्यो होलिन जुन सिक्वेन्सिङ गर्नुभयो, त्यो होलिनको त तपाईले एक्टिभिटी हेर्नु भएन नि है?
Sorry sir, I didn't get it.
Sorry, I'll put it in English. So I assume that you did the cloning of the holine from one of the bacteriophages you isolated.
Oh, sir.
But then again, like I don't think you ever isolated in protein form, and did you check for the antibacterial activity of the holine protein, not the bacteriophages, on your Klebsiella?
Yes, exactly. We have collected carbapenem resistant Klebsiella pneumoniae from those patients who are causing UTI.
I'm more interested on the protein that you have cloned into DS5 alpha. did you actually isolate the protein? I'm not talking about the bacterial phase now.
Yes, sir.
So I'm talking about the choline protein that you have cloned into E. coli. Did you isolate the choline protein and check its antibacterial effect on your bacteria?
No, sir.
No?
Actually, we have identified choline gene from that phage protein genome, and then that choline gene was ligated with the compatible vector like DH5 alpha, and then after that ligated or recombinant that vector is transformed into the competent cell, sir.
Yeah, I get that. And I got one more question is, did you ever see resistance to these bacteriophages or the product that you are sequencing? What about the resistance to the bacteriophages that you're using? Did your Klebsiella pneumoniae develop resistance to the bacteriophages when you were co-culturing them?
No, sir. Klebsiella pneumoniae, carbapenem-resistant Klebsiella pneumoniae, against that we have isolated phage. Yes, and after isolation of that phage, from that phage we have extracted the DNA, and then from that genome we amplified the choline gene, because we have already performed the whole genome sequencing of that phage.
Okay, thank you very much. In my experience, I work with a little bit in Salmonella typhi and Paratyphi, and also a little bit of Klebsiella. So when you are co-culturing the bacteria with the bacteriophages, they kind of control until 6 hours, but then again when you come back next day, when you put both of them, the next day these bacteria all grow to as much as the controls. So they actually develop resistance by the next day. So,
Okay,
thank you.
thanks.
I got your answer. He didn't, I think, understood your question also. Choline, as a choline, lots of paper are there. Choline is responsible for the killing of those bacteria. And so on the basis of that, we have focused on the choline genes, okay, sir? And whole genome sequencing after, the choline was identified with different techniques, and then we haven't pure choline till now, okay, sir? We have to express it in vectors also. This still the research is going on. Maybe in 1 or 2 months we will do this. And your next question is resistance develop or not? Yeah, sure, resistance will be developed because CRISPR-Cas will be produced by bacteria to combat the viruses in bacteriophage. So sure, after 3, 4, 5, when we keep it for old, then we see the growth of the bacteria in between. That's right, you are right, sir. We have also that kind of result, and in some case we couldn't see like this. Thank you very much.
Any more questions? Okay, now we'll move on to the next presentation. Thank you very much. So our next presenter is Sivaram Naga, who has been serving as a clinical research coordinator at Dhulikhel Hospital since November 2014, accumulating over 9 years of dedicated service in this role. Describing himself as a healthcare servant, compassionate leader, Mr. Siva embodies qualities of mentorship and coaching, guiding his peers towards professional growth and excellence. Over to you, sir.
Thank you, chairs, and I also thank NHRC for giving me a platform to present my work, and warm evening to all. Today I am going to present on school as a platform for a rapid typhoid seroepidemiologic assessment. I am Sivaram Naga from Dhulikhel Hospital. Sorry. Okay. As we all know that Nepal has a high typhoid burden in the Kathmandu, particularly in the Kathmandu Valley, with the sero incidence of 330 per 100,000 person year. And in the other part of country, particularly in the rural area, there is a limited evidence of typhoid in other rural area of Nepal. And we know that conducting blood culture surveillance is valuable, but it's challenging. And it is not only the expensive, but logically intensive too. And it often takes many years to complete. So in light of this challenge, sero survey emerged as an alternative approach to generate an accurate typhoid incident estimate. So moreover, we propose a school as a potential alternative to the quick estimate of typhoid burden, but it is unclear that whether they provide the representative samples for assessing community exposure of Salmonella typhi or not. So the main objective of our study is to determine the feasibility of using school-based sampling frame for the typhoid seroepidemiology and evaluating the participant rate, research requirement for conducting study, and also to compare the school-based and population-based sero-incidence estimate from a same community to determine whether school-based sampling estimate provide the unbiased estimate with the population-based survey. So for our study, we collect a sample from randomly from 18 schools, primary and secondary from Dolkha and Kavri district. And we collected sample from 4 municipalities in the Kavri district, Panoti, Banepa, Tulikhel, and Panskal. From each municipality, we collected 1 public and 1 private school for school-based sampling. And from the Dolkha, we select randomly selected sampling, which includes 1 private schools and 9 public schools. And we selected up to 100 children from each randomly selected schools. And we have certain criteria for our study that the students should be aged between 4 to 18 years and also provided a consent by their parents. And for our data collections, we collect a capillary blood, which we collected in a filter paper. As we saw, you can see in the picture, it's unclear. We have a dried blood spot, which we collect samples by pricking finger pricks. So we collected a very few samples from the students. So it is also less invasive. And for our analysis, we did measure the HLYE IgG and HLYE IgA levels, which we measured by using kinetic ELISA technique. And we have already published the population's data from 2019 to 2020. And this is the sample characteristics of the students in the school study, school-based study. We did the school study in Dolkha and Kavre, and for population study, we did in Kathmandu and Kavre. So we have the same catchment area for Kavre, and we measured the sero incidence in age group 5 to 15 years, and we can see that both of Dolkha and Kavre has the male and female ratio is equal in both Kavre and Dolkha district. And this table shows the characteristic of school and population-based samplings. We did the population-based sampling in 2019 to 2020, and the school-based survey in 2020 and 2021 and 2022. And both of we have the same catchment area in Kavre that we collect a sample from 4 municipalities in Kavre district, and the median age is similar, but the sample size is lower in the population-based samplings. But the durations of conducting study is 24 months, which is 2 years, and the research staff required for conducting study is high compared to the school-based survey. If you see the participant rate is higher in the school-based sampling. So we determined the sero incidence by determining we adapted the longitudinal immune response of HLYE IgG and HLYE IgA among the culture confirmed enteric fever cases for 2 years, and we used the antibody dynamics to calculate the sero incidence in a population, cross-sectional population-based data. And so we calculate sero incidence in Kathmandu, Kavre, and Dolkha district. We found that the sero incidence is very high in Kathmandu, and we also found that the sero incidence derived from the Kavre school-based sampling is very much lower than the that one derived from the population-based sample. And as expected, the rural region of Nepal, Dolkha, has a very low sero incidence. We also observe in the municipality level that the sero incidence in Kathmandu is higher, and is of the 4 municipality of the Kabri district. We observed that the sero incidence derived from the school-based sampling is much lower than the sero incident derived in the population-based samplings. But interestingly, we found that the 2 municipality in Dolka, that is Bigu and Boitesur, has a higher sero incidence, which is comparable to the sero incidence in a Kabri school-based sampling. So, in conclusion, we can say that the sero incident estimate derived from the school-based samplings were lower than those derived from the household survey, but the participant rate were higher in the school-based survey in comparison with the household survey, and the sampling was able to perform within the fraction of times and with the fewer personnel, so which will markedly reduce the cost of the study. Also, since school-based survey still could be enabled rapid mapping of typhoid risks in a community where the blood culture surveillance is not available, as well as to monitoring the typhoid exposure which change following the vaccine introduction. Lastly, based on our finding, it can be inferred that Dolka, a rural district of Nepal, exhibits a low incidence rate and bear a significant reduce of burden of typhoid. I'd like to acknowledge my entire team for their collaborative effort. Thank you.
Very much.
So, any questions?
Okay, now floor is open for the question.
Thank you once again. It was a nice presentation. My question was, during your presentation, you said that the culture confirmed cases were tested for the sero incidence.
Yeah, we did longitudinal antibody response in 2019 to 2021. We have different sites in Kathmandu and Dhulikhel Hospital. There is 6 sites in Kathmandu, Bir Hospital, Nepal Medical College, and Helping Hands, Alka hospitals. We have different hospitals that we collect culture confirmed case, and we follow up to 2 years to determine the antibody response from them. We collect a sample at 4 point at baseline, 28 days, and 3 months, and 6 months, and the last visit. And we determine the antibody decay on that.
So you also talked about IgG as well as IgA.
Yeah.
So if we can differentiate it by some other method than ELISA, it would be easily available for screening later. So at present, you're doing the kinetic ELISA, and it would be differentiated IgG as well as IgA.
Yeah.
So was there any difference in the culture positive cases?
Sorry?
You said that IgG might wane, but what about IgA? Was there a significant difference in the antibody levels, is what I was trying to ask. Or it is a cumulative ELISA which will give
it's not like it's a cumulative. We all collect all data from all patients, and we did. Actually, it's a cumulative work, not the individuals and source.
No, the antibody. Can you differentiate it as IgG as well as IgA? Are they detected separately or as a cumulative antibody?
It's a separate. We determine separate IgA and IgG.
So was there any significant difference is what I was asking?
Sorry, the paper is done by the Christians, one of our collaborative. I'm sorry.
Thank you.
Any more questions?
Thank you for the nice presentation. I also work with the typhoid thing, so I'm interested in your work. My question is, why did you choose this hemolyzin E as an antigen? Because is there any specific reason for choosing it?
Yeah,
and what kind of ELISA is this? Is this RDT or some kind of like?
It's not RDT. It's plate-based.
Plate method.
Yeah, we create antigens ourselves. Previously, we choose 5 different antigens: LPS, HLYE, and MP. And VI, and we found that SLYZ has a consistence and high titer. We choose two previously, LPS and SLY, but LPS is didn't show up good in case of Salmonella paratyphi A, and LPS has also very like non-specific reactions with others non-salmonella typhi. So we choose SLY for estimating the sero incidence.
We are also interested in doing similar kind of work with our stored plasma samples. And my question is, like we can see the IgG and IgA, but not IgM. Normally for the active active infection, we look for the IgM. So you're doing the IgA. So is IgA?
We did a lot of data survey. IgA also, IgA also give a good marker for acute infections. That's why we choose the IgA.
Okay. My last question is that did you purify the antigens here, or were you able to access it with some donations?
For this study, particular study, we did send to the outside the country, MCS, for the analysis, and before we get purified antigens for our study.
Thank you.
Thank you.
Thank you very much. So we'll move on to the next speaker. Our ninth speaker of this session is Dr. Noadat Subedi, who is an accomplished associate professor of forensic medicine with over 11 years of dedicated experience in research and academics. Currently working as principal investigator in mortality surveillance program entitled Minimally Invasive Tissue Sampling to Strengthen Identification and Characterization of Cause and Perinatal Death in Kaski district of Nepal. Over to you, sir.
Thank you so much. Thank you, organizers, for the opportunity to present in this conference. Can we get the slides? This is an ongoing project where we have implemented minimally invasive tissue sampling to the existing maternal and perinatal death surveillance and response system in the Kaski district of Nepal. So it's collaborative work done by all the MPDSR implementing hospitals in Kaski district of Nepal. Sorry for that interruption. यो MPDSR सिस्टम चाहिँ फ्यामिली वेलफेयर डिभिजनले सेलेक्टेड हस्पिटलहरुमा गरिराखेकै नै छ। खास यसको मेन अब्जेक्टिभ भनेको टु आइडेन्टिफाई द कजेस अफ डेथ and to review the causes of death so that some response can be implemented for the reduction of similar death in the future. तर यसमा चाहिँ यो MPDSR system implementation गर्न there are several challenges, and one of the most important challenge is the cause of death identification is difficult at times. हामीले अहिले recent family welfare division को MPDSR fact sheet हेर्यौं भने 53% of the stillbirth मा cause of death केही पनि भेटिएको छैन र 8% of the neonatal death मा cause of death भेटिएको छैन। जब हामी cause of death भेट्दैनौं तब response हामी ड्राफ्ट गर्न सक्दैनौं। त्यसैले यो MPDSR सिस्टममा चाहिँ cause of death एक्सप्लोर गर्न इम्पोर्टेन्ट भएको हुनाले हामीले जुन एक्जिस्टिङ सिस्टममा एडिसनल्ली मिनिमली इन्भेजिभ टिस्यु स्याम्पलिङ युज गरेर टु डिटरमाइन द कज अफ पेरिनाटल डेथ बाई इन्टिग्रेटिङ मिनिमली इन्भेजिभ टिस्यु स्याम्पलिङ इन पेरिनाटल डेथ द्याट आर रिभ्युड अन्डर हस्पिटल बेस्ड MPDSR इन कास्की डिस्ट्रिक्ट। यसको साथसाथै हामीले चाहिँ कुन टिस्युले बेटर आइडेन्टिफाई गर्छ द कज अफ डेथ भनेर पनि अर्को अब्जेक्टिभ राखेको छौँ। So it is a cross-sectional observational study र यसको स्टडी पपुलेसन चाहिँ कास्की जिल्लामा MPDSR इम्प्लिमेन्ट गर्ने 5 वटा हस्पिटलहरुमा हो र यसमध्ये 3टा चाहिँ मेडिकल कलेज र युनिभर्सिटीहरू छ, गण्डकी मेडिकल कलेज, मणिपाल कलेज अफ मेडिकल साइन्स र पोखरा एकाडेमी अफ हेल्थ साइन्स, र 2टा प्रोभिन्सियल हस्पिटल, मातृ शिशु मित्री हस्पिटल र शिशु हस्पिटलमा हामीले यो कोलाबोरेटिभली इम्प्लिमेन्ट गरिरहेको छौँ। र यसलाई हामीले कसरी इन्टिग्रेट गर्छौँ त भने जुन एमपिडिएसआर सिस्टम छ यो गभर्मेन्ट प्रोजेक्ट हो, यो चलि नै राखेको छ। यसमा चाहिँ क्लिनिकल डेटा, क्लिनिकल र डेमोग्राफिक डेटा अफ द पेरिनेटल डेथ इन्क्लुडिङ स्टिल बर्थ 28 हप्ताभन्दा माथिको र जन्मेर 7 दिनभित्रको नियोनेटल डेथमा चाहिँ मदरको र त्यो चाइल्डको क्लिनिकल इन्फर्मेसन ग्यादर गरेर एउटा स्पेसिफाइड फर्म छ त्यो भरिन्छ र त्यो त्यसमा भरेर महिनामा एउटा मिटिङ हुन्छ, त्यसको एनालाइसिस गरिन्छ। हामी त्यही हस्पिटल एमपिडिएसआर कमिटीसँगै इन्टिग्रेट गरेर एडिसनल टेस्ट गर्छौँ, जुनमा स्टिल बर्थ र अर्ली नियोनेटल डेथमा हामीले मिनिमली इन्भेजिभ टिस्यु स्याम्पलिङ गरेर टिस्यु स्पेसिमेनहरू निकाल्छौँ र डिफ्रेन्ट टेस्टहरू गर्छौँ, हिस्टोप्याथोलोजी, माइक्रोबायोलोजी, हेमाटोलोजी, पिसिआर टेस्टहरू गरेर सेपरेटली कज अफ डेथ आइडेन्टिफाई गर्छौँ। साथसाथै हामीले चाहिँ एक्जिस्टिङ प्लेटफर्मबाट पनि इन्फर्मेसन लिन्छौँ र त्यो हामीले निकालेको कज अफ डेथको डेटा हामी एमपिडिएसआर कमिटीलाई पनि सेयर गर्छौँ र कुनै एडिसनल रिकमेन्डेसनहरू छ भने त्यो हामी सेपरेटली पनि दिन्छौँ। सो इट्स नट ओन्ली अ रिसर्च, सो इट्स अ प्रोजेक्ट इन्टिग्रेटिङ विथ द एक्जिस्टिङ एमपिडिएसआर सिस्टम। So talking about the sample collection, we take nasopharyngeal swab in the neonatal death. हामी स्टिल बर्थमा लिँदैनौँ र CSF र स्टिल ब्लड लिन्छौँ हामी बोथ नियोनेटल स्टिल बर्थमा। र टिस्यु स्याम्पलको कुरा गर्दा चाहिँ हामी ब्रेनबाट, लङ्स र लिभरबाट स्पेसिफाइड बायोप्सी निडल युज गरेर हामी टिस्यु स्याम्पल एक्सट्र्याक्ट गर्छौँ। र स्टिल बर्थमा चाहिँ हामी प्लेसेन्टा एक्जामिनेसन पनि गर्छौँ। प्लेसेन्टा एक्जामिनेसन रुटिनली गरिँदैन र एज अ पार्ट अफ दिस प्रोजेक्ट हामीले प्लेसेन्टालाई पनि एक्जामिनेसन गरेको छौँ। र स्याम्पल एनालाइसिस हेर्दा चाहिँ नाजोफरेन्जियल स्वाबमा हामी ग्राम स्टेन र ब्याक्टेरियल कल्चर गर्छौँ र पिसिआर पनि गर्छौँ। प्लेसेन्टल स्याम्पलको लागि हामी हिस्टोलोजी र ब्याक्टेरियल कल्चर गर्छौँ। ब्लडमा हामी ब्याक्टेरियल कल्चर, सेरोलोजी, टर्च स्क्रिनिङ, ब्लड ग्रुपिङ, आइरिस टाइपिङ र मलेरिया स्क्रिनिङ गर्छौँ। र सिएसएफमा माइक्रोबायोलोजी र सिएसएफ एनालाइसिस गर्छौँ। ब्रेन टिस्यु, लङ्स र लिभरमा हामी हिस्टोलोजी गर्छौँ भने लङ टिस्युको हामी कल्चर पनि गर्छौँ। र यसरी हामीले निकालेको स्पेसिमेन र यसबाट आएको रिजल्टहरूलाई हाम्रो कज अफ डेथ प्यानल हामी सेपरेटली बनाउँछौँ, जसमा स्टिल बर्थमा कम्पल्सरीली अब्स्टेट्रिसियन हुनुहुन्छ भने नियोनेटल डेथमा नियोनेटोलोजिस्ट हुनुहुन्छ र अरू टिम मेम्बर पनि भएर सबै एभाइलेबल डेटालाई हामीले कम्पाइल गरेर आइसिडिपिएम जुन WHO को रिकमेन्डेसन छ, आइसिडिपिएम अन्तर्गत हामी कज अफ डेथ डिराइभ गर्छौँ। र त्यसको साथसाथै रिकमेन्डेसन टु प्रिभेन्ट द फ्युचर डेथ, प्रत्येक केसको हामी केस समरी बनाएर हामी प्रत्येक केसलाई कसरी मिनिमाइज अथवा प्रिभेन्ट गर्न सकिन्थ्यो भनेर रिकमेन्डेसन पनि दिन्छौँ। इट्स अनगोइङ स्टडी, यो 3 इयर प्रोजेक्ट हो र हामी लगभग 1 वर्षको अन्त्यतिर आइपुगेको छौँ। त्यतिमध्ये 162 डेथ नोटिफाइड भएको मध्ये 135 मा हामी कन्सेन्टको लागि एप्रोच गऱ्यौँ। देयर वाज फेयर कन्सेन्ट रेट, 80% कन्सेन्ट रेट अब्टेन्ड। र मैले यो प्रोजेक्टको जस्ट स्यालेन्ट फाइन्डिङ मात्रै प्रेजेन्ट गर्न चाहन्छु। हामी अर्ली डिसेमिनेसन किन गर्दैछौँ भने जुन एमपिडिएसआरलाई सुरुको स्टेजबाटै स्ट्रेन्थन गर्न सक्यो भने यसले चाहिँ बेटर एभिडेन्स दिन्छ र प्रिभेन्टिभ मेजरहरू पनि एडभोकेट गर्न सकिन्छ भनेर यसमा हामीले कज अफ डेथ मिट्स गर्नु अगाडिको कज अफ डेथलाई हामी यो ग्रे कलरमा प्रेजेन्ट गरेको छौँ भने मिट्स एड गरिसकेपछि ब्लू कलरमा प्रेजेन्ट गरेको छौँ। 8 वटा अनस्पेसिफाइड, अहिलेसम्म हामी 73 केसको डेटा कम्पाइल गरेर प्रेजेन्ट गरेको हो। इन 8 केसेस देयर वेयर नो एनी कज अफ डेथ आइडेन्टिफाइड, बट आफ्टर इन्टिग्रेटिङ मिट्स सबै केसमा केही न केही कज अफ डेथ हामी भेटेको छौँ। त्यस्तै ग्रोथ डिसअर्डर 10 मा लेखिएको थियो बिफोर इम्प्लिमेन्टेसन अफ मिट्स भने हामीले ग्रोथ डिसअर्डर इज नट ए स्पेसिफिक कज अफ डेथ। whenever we don't get other cause of death, we can write growth disorder, so that decreased from 10 to 2. Likewise, the intrauterine hypoxia we could diagnose in 31 cases, whereas before MITS it was diagnosed only in 18 cases. Likewise, syphilis, any intraamniotic infection which could never be diagnosed as a routine MPDSR system, we could identify through the MITS. लाइकवाइज म्याटर्नल कज अफ डेथमा आइसिडिपिएममा फिटल कज र म्याटर्नल कज दुवै लेखिन्छ। म्याटर्नल कज हामीले कुनै मदरको एडिसनल टेस्टहरू गर्ने त होइन, तर हामी प्लासेन्टल एक्जामिनेसन गर्ने हुनाले जुन प्लासेन्टल एब्नर्मालिटी बिफोर वी युज दिस इन्टरभेन्सन इट वाज ओन्ली इन 2 cases इट वाज डायग्नोज, बट हाम्रो इन्टरभेन्सन पछि 20 केसमा प्लासेन्टल एब्नर्मालिटिजहरू डिटेक्ट गर्न सक्यौँ। त्यसै गरी इन 13 cases देयर वेयर नो एनी म्याटर्नल कजेस आइडेन्टिफाइड, but it reduced to 5 after the MITS. Likewise, chorioamnitis हामी 9 cases diagnose गर्न सक्यौं र medical and surgical condition it decreased from 33 to 15 because we could diagnose more specific causes of death. So in 73 cases of perinatal death हाम्रो यो MITS add गरिसकेपछि 79% मा केही न केही यसले फाइन्डिङ दियो, भनेको cause of death मा यसले डेटा प्रोभाइड गर्यो र कुन टिस्युले बढी फाइन्डिङ दियो त भन्दाखेरि प्लासेन्टल हिस्टोलोजी इन 30 cases, बट इट इज अन्डर इस्टिमेसन। किन भन्दा हामीले प्लासेन्टल हिस्टोलोजी प्लासेन्टल एक्जामिनेसन स्टिल बर्थमा मात्रै गर्छौँ, इट इज एग्रिगेट अफ नियोनाटल एन्ड स्टिल बर्थ्स। So placental histology is very, very important to identify the causes of fetal death. Likewise, lung histology, blood microbiology, nasopharyngeal microbiology, lung microbiology, placental microbiology can give the causes of death. In 46 cases of stillbirth among the 73 that we have presented, 76% मा हामी placental examination complete गरेको थियौं भने त्योमध्ये 94% मा चाहिँ placental examination ले केही न केही cause of death add गरेको थियो। र the recommendation को कुरा गर्दाखेरि antenatal care should be improved in our context. There are many shortcomings with the antenatal care. Likewise, health education, health seeking behavior and others which have been identified by our decode panel members, we have mentioned here. So यो हाम्रो इनिशियल फाइन्डिङ हेर्दाखेरि हामी के देख्यौं भने जुन एक्जिस्टिङ एमबिडिएसआर मा कज अफ डेथ पत्ता लगाउन गाह्रो भइरहेको थियो। जब हामी कज अफ डेथ पत्ता लगाउँदैनौं, रेस्पोन्स फ्रेम गर्न सक्दैनौं, त्यही भएको भएर केही इन्टरभेन्सन गर्न जरुरी छ हैन। मिट्स क्यान बी इम्प्लिमेन्टेड इन द एक्जिस्टिङ एमबिडिएसआर सिस्टम टु स्ट्रेन्थन दिस डेथ सर्भिलेन्स सिस्टम। तर वी क्यान अन्डरस्ट्यान्ड द लिमिटेसन। जुन एक्जिस्टिङ एमबिडिएसआर चलाउनलाई पनि अब बजेटरी कन्स्ट्रेन्टहरू छँदैछ, तर त्योमध्ये सबभन्दा मिनिमम स्याम्पलहरू कसले कज अफ डेथ आइडेन्टिफाई गर्न सक्छ भनेर हाम्रो इनिशियल फाइन्डिङबाट हेर्दा प्लासेन्टल एक्जामिनेसन जुन कुरा चाहिँ प्लासेन्टा इज जस्ट डिस्कार्डेड आउट विदाउट एक्जामिनेसन, सो इट सुड बी एक्जामिन। ग्रस एक्जामिनेसन अथवा मेजरमेन्ट मात्रै भए पनि गर्नुपर्ने देखियो। लाइकवाइज अब लङ ब्लड र एडिसनल स्याम्पलहरू गर्न सक्यो भने त अझै केही न केही एडिसनल फाइन्डिङहरू दिन सक्यो। सो कन्क्लुडिङ, मिट्स क्यान प्रोभाइड एडिसनल एन्ड स्पेसिफिक कज अफ डेथ। सो जुन मिसक्लासिफाई भइरहेको थियो हामीले कुनै पनि टेस्टहरू नगरेर कज अफ डेथ लेख्दाखेरि, सो मिट्स एड गरेपछि जुन मिसक्लासिफिकेसनलाई यसले घटाउँछ र मोर इफेक्टिभ रेस्पोन्स फ्रेम गर्नको लागि यसले धेरै इम्पोर्टेन्ट रोल खेल्छ। र हाम्रो कन्टेक्समा चाहिँ एन्टिनेटल केयरको कमी देखियो। कति मदरहरू जुन रिकमेन्डेड एन्टिनेटल केयरको नम्बर मात्रै नभएर क्वालिटी अफ एन्टिनेटल केयरको पनि हाम्रोमा कमी देखियो। So the ways forward, we are working on generating the evidence and the usefulness of myths. So whenever we get the evidence, we'll start to advocate to strengthen the existing death surveillance system. So I'd like to acknowledge our collaborators, Myth Surveillance Alliance and several collaborators. It was funded by the Myth Surveillance Alliance, which was further funded by the Bill and Melinda Gates Foundation. Thank you very much.
Thank you very much. The floor is open for the question. I think there are no questions. Thank you very much.
Thank you, sir.
So now we'll open the floor again for the first presenter, thematic paper by Professor Dr. Prakash Kimire. If there are any questions for him, we'll take 2 straight questions. I think everyone is hungry. Sir.
यो धेरै सर्ट छ यो। सो आई कङ्ग्रेचुलेट प्रकाश सर फर हिज एक्सटेन्सिभ प्रेजेन्टेसन अन इन्फेक्सियस डिजिजेस विच इज अ वार्निङ प्रोग्राम। And another thing is I want to just give one information regarding the International Society for Infectious Disease Conference, which is going to be held in December 6 onward. And I request all our young scientists to send their abstract, and those good abstract from the low-income countries like ours, few will be also rewarded with the traveler grants, and also it will be published in the International Society for Infectious Disease Journal. So it will be a kind of some sort of research encouragement for young scientists. Thank you.
Thank you very much, sir. So we are at the end of the session. So I would like to request Professor Dr. Jeevan Sherchand sir to give his remark.
So now we are at the end of session. So I would like to congratulate to all presenters who have presented and dedicated their work in front of our audience. So I think a number of presenters have presented very innovative research findings, which is, I think, one of the very initiation is a part of the infectious disease and microbiology. So at this end, again, I would like to congratulate to all of presenters and also all listeners, audience, scientists, and our health-associated researchers. Thank you very much indeed.
Thank you very much. So yeah, we have come a long way and we have successfully completed our 8th session of this 10th National Summit. It was really appreciable for all the speakers. And now, having said that, I would like to request again all the speakers to come on the stage for a group photo.